Chancellor, Professor Dhrubajyoti Chattopadhyay, sir, to come upon the stage and kindly address our student and the audience. Thank you, sir.
A very good afternoon to my dear students, the faculty members, and our guest speaker also over here. Now today the whole program is designed in such a way that it has got two parts. The first part is very important. The students who have actually visited the Sakura Science program in Okayama University and did their work for 3 weeks. They are going to present their experience, what they have done, and what they feel about it. So I think a couple of students who are going to present first, and then we are going to have another lecture, and that's why it's named as Industry Connect, from a person who is working in the industry, specifically pharmaceutical industry, for a long time, and thank you for visiting us and coming over here. Actually, Shobhoshaji could not remember, but I remember his work when he presented in Bush Institute, actually. Professor Pradeep Parekh is my friend, who is his PhD guide. And so from those Bush Institute time, because I worked my PhD in Lambda, his work is also in Lambda. That's why I felt very excited about it. But later on, he moved and did some very elegant work abroad. But after coming over here, the specific topic, what he's going to discuss with you, my dear students, these are very important because during the COVID period, there is quite a bit of discussions about this biosimilars and other things. Now, actually, what is actually its impact, what it means, and how it's going to work out in that particular area of biopharmaceutical industry, I'm sure Professor Haldar is going to discuss with you. And with this brief introduction and also welcoming all of you over here, thank you very much.
Thank you, sir. Now I would request Dr. Rajat Pal, Assistant Director, Student Affairs of Sister Nivedita University, to come upon on stage and kindly introduce our students who have received Sakura grant, Science grant, and have been there in the Japan for 3 weeks. Please, sir, come and introduce them.
Good afternoon, everybody. So this is a very big opportunity, I'm saying, that the students to whom I am going to introduce, they are probably the gem of this university and this department. So I'm very glad to introduce my very good students who have been fortunate enough to visit Japan for having or availing the Sakura Science grant to the Okayama University. Where is Surjit? Yeah. I need to thank Surjit because he has taken the whole pain for having these 7 kids with him for at least 3 weeks, I guess. Yeah? Exactly. Okay. So it's an opportunity for him. Yes. So, I am just reading the names. The students who have got this fellowship. Shopnonil Pal, Shakhar Shom, Ritom Pal, Odrija Bashu Roy Chaudhuri, Piyasha Mukherjee, Prantika Maiti, and Srija Shau. So I guess a very congratulations to all of them that they have availed this grant and they have done a wonderful job, what feedback I have got. And they have gathered quite a bit of knowledge regarding their coming scientific career. So I'm again requesting Shani to please conduct that session So once again, congratulations to them.
Thank you, Rajat. Thank you, Rajat sir. Now we are moving to our first session. So, as our first presenter, I would request Swapno Neil to come upon stage and present his work entitled "Involvement of CABL in Regulation of AIDS, a B cell AID, a B cell genome mutator." By Swapno Neil Pal. Please.
Hello. A very good afternoon to everyone present here, to all the delegates, to the speaker, and my classmates, and my juniors. So, today, I am going to present the short research progress that I did in Okayama University at the Molecular Cell Engineering Lab led by Dr. Kanae Manauki. So, in my work, what I did was, we tried to understand the regulatory mechanism in the B cells, in the B cell development, which leads to high affinity antibody production. We know that the lymph nodes are divided in these germinal centers, these centers that are required for development of the B cells. We have the mantle zone, the dark zone, and the light zone. These dark zones are the areas where the B cells get the mutation, they acquire the mutation, somatic hypermutation as it is known, and that leads to high affinity antibody production, right? Now, this is the whole process of B cell development. You can see when the B cells reside at the dark zone, these B cells are known as centroblasts. After acquiring these mutations at the variable region of the antibody, they convert into centrocytes. The overall concept of somatic hypermutation is well known by now. So it is basically nothing but the mutation that B cells get in their genome through a particular enzyme known as activation-induced cytidine deaminase, shortly known as AID, at the variable region again. At the nucleotide level, what AID does is it basically deaminates the cytidine nucleotides, and that leads to formation of uridine. Now, uridine is not a natural base that should stay in DNA. So eventually it gets cleaved out, and that leads us with these mutations, these point mutations. The whole mechanism of SHM is quite complex, so I'm not going to in that detail, but AID acts as a key regulator, as a key player in that process. Now, since AID is a very important enzyme, it is mutating the DNA, so there should be multiple levels of regulation that AID should be controlled with. And there are multiple levels of regulations. We can see there are transcriptional level regulation, there is post-transcriptional level, there are particular miRNAs that regulate the expression of AID. At the post-translational level, there is high level of regulation that happens because AID is not expressed in other cells, the cells other than B cells, right? And especially B cells also, when B cells are in the dark zone, only at that time AID is expressed, because if it is unregulated, that can lead to certain types of cancer. At the Molecular Cell Engineering Lab, what the workers have elucidated so far is this axis of regulation of AID. Now, this part is well known. Now, there could be other key players in the activation of AID. They have found out that there is a negative regulation axis of AID. So basically, if we knock down this LIN, which is again an upstream regulator of AID, if in the knockout background cells, DT40 cells in this background, we can see that AID is highly expressed as anticipated because it is a negative regulator. Upon introducing a particular SRC kinase inhibitor, PP2, a small molecule, that can again lead to downregulation of AICD gene. In the BCR-KO, as I said earlier, since this is a negative regulation of AID, and since we anticipated that, okay, probably it is BCR-dependent pathway, so upon knocking out the BCR, what we can see is the AID expression is downregulated. So this axis is well established by now, that PP2 inhibits LIN, and eventually this B cell dependent pathway, this is the way that AID is regulated. Now, there are other important candidates. So one of the candidates that the lab found out was that c-Abl. C-Abl is another SRC kinase, but this is a positive regulator. How? In the c-Abl knockout background, what we see is the expression of AICDA goes down, right? And again, upon the introduction of PP2, the levels of expression of AICDA goes up. So this is quite intuitive that there are two axes of regulation. One, c-Abl could be involved in the positive regulation, and this LIN-regulated pathway, the negative regulation is there. Now, for the further work, what I did was I amplified the c-Abl transcripts from the cDNA and purified these amplified products, and I prepared the vectors for cloning, which was SRSF1 delta RAR vector. So this is the clear band that we can see, 3.5 kilobase pair, which is one of the isoforms. There are multiple isoforms present in the whole transcriptome of B cells for c-Abl. So that was the isoform 1. And for the restriction digestion, we can see the empty vector at 9 kilobase pair, and the insert sequence is free. So that is how I prepared these empty vectors for further cloning of this c-Abl gene, isoform 1 of it. So in conclusion, I have purified this isoform 1 of c-Abl, the CDS of that, and the SRSF1 delta RAR vector will be used for further expression of c-Abl in TT40 cells and for further studies. So in future, what we can look at is how these other regulatory, upstream possibly regulatory, candidates, how they act on AID. Is it phosphorylated or some other, regulatory, mechanisms are there? So that can be checked, and this small segment that I did can be important in that purpose. So finally, thank you to every single person involved in the Sakura Science program for making this happen. I would wholeheartedly thank my supervisor at Okayama, Dr. Kanayama Sensei. Without him, the research wouldn't be possible. I would also like to thank the senior, Michiro-san, who helped me throughout the process. And again, thank you so much. So if there are questions, you can ask. You can go ahead.
So I have a question. So, do this thing have any translational value? Are they going to apply in a translational medicine?
Right. So, as I said earlier, the small molecule that we are using, PP2, it is, again, SRC kinase inhibitor. That is one aspect of it. Now, if we actually get to know the axis that I talked about, the positive regulatory mechanisms, if there are key, checkpoints that are important, that is BCR independent, because if this axis that we are looking at, the negative regulatory axis, if this cannot be addressed in a cancer, in a B-cell lymphoma, what we can do is we can look at particular small molecules that can act on the upstream regulators of the positive regulatory mechanism. That can be an important aspect of this.
Is this process anyhow affecting antibody production or BCR? You know that B cell is also used, B cell is also have antigen presentation property.
Yes.
So is it affecting this property of B cell?
No.
Or they have any other antibody production, memory cells generation?
No, till now, memory cell generation, that aspect has not been looked at, and the antibody, the antibody presentation, that is also has not been found out yet. We're just finding out the candidates that can be involved in the positive regulatory, the axis of AID.
Thank you.
No questions? All right.
Okay. Next presenter. Thank you, Shopin. Okay. So now I would like to call Ritam and Shakhar Shom, who will be presenting their work which they have done in Japan. And their title of their work is Production and Characterization of the RO52 Antigen Antibody System and PDCD1 Immune Checkpoint Gene Cloning.
Good afternoon, everybody. So, as you can understand from this first slide, cover slide itself, there is something that is tangled and we worked on to untangle it. Now, before I dive deep into the topic, I like to introduce myself. I am Shakhar Shom, a master's final year student at SNU, and I got the chance to engage in the Sakura Science Exchange program at the medical protein engineering lab in the lab of Junichiro Futami. And now, coming back to the topic, so as I mentioned before, something is tangled. Now, what is the tangled thing? The tangled thing here is the RO52. What is RO52? RO52 is a E3 ubiquitin ligase which acts on antibody-bounded antigen, and it pushes the antigen to E3 ubiquitination through proteasome digestion. Now, what is the main problem? Because RO52 is produced inside the cells. So, in cancer cells, this RO52 protein, due to cancer cells having a leaky vasculature, as we know, this RO52 tends to leak into the blood vessels, and this RO52, when leaked, is acted upon by B cells which produce antibody against the RO52, which is the anti-RO52. Now, we understood the mechanism. Now, what is the problem when we try to replicate this in the lab? In the lab environment, when RO52 is produced from bacterial cloning systems, the protein tends to get tangled and aggregated and form a precipitate. To overcome this, and suppose we take imagine a ball of wool, and you will try to make some sweater or anything out of it. You cannot work with it when it is a ball. You need to entangle it like a string. Then you try to sew the sweater. Similarly, how to entangle the RO52? We used a mechanism that is the TAPS sulfonate solubilization. What TAPS sulfonate does is it attacks the cysteine-rich residues and adds a TAPS sulfonate group to the cysteine-rich residues, which eventually gives a positive charge to the protein, and due to positive charge repulsion between the proteins, the protein do not tend to get aggregated, and as a result, they get solubilized in the water solution and in the aqueous phase. Now, this is the basic workflow. We lysed the cells, then took the antigen, that is RO52. We added TAPS sulfonate. Then, for purifying the protein, we used dialysis method to get the pure antigen. These are the results. In the UV analysis, the peak at 280 nanometer, sharp peak, you can see, we can confirm that RO52 was present. And also in the HDS page analysis, as you can see in the TAPS RO52, TAPS treated RO52, there is a distinct broad band at the supernatant, SUP. So, if it is soluble, it will never give a band in the precipitate. It will give a band in the supernatant. This clearly signifies that our designed RO52, our produced RO52 was solubilized in water. There are some traces in the precipitate which could not be captured, but this is the whole thing. Now, to check that we produce the right antigen, we also had a cloned antibody and anti-RO52 in the bacterial cells. We produced the anti-RO52, then purified it using cobalt NTA column through affinity chromatography, and after each elution, you can see the peak in the spectro, it reduces. So it showed that we significantly collected, purified the anti-RO52, and now for checking it, we did the Western blotting. We run SDS gel, and in the Western blotting, at different concentrations of RO52, even at 0.625 microgram per microliter, it showed a band showing that the sensitivity of the antigen and the antibody interaction. And so, where does this system actually work? We synthesize the antigen, antibody, everything, but where does this system actually work? So, there is a system called the Muscat assay. That is, there is a bead, immobilized bead, where we add the S-catenized antigen, and when this metal bead is injected into the patient's body, the autoantibodies, the anti-RO52, act upon these metal beads, and when treated with the secondary antibody attached to a probe, they give a fluorescence signal, which signifies that there is a high chance of presence of anti-RO52, signifying the patient is undergoing through cancer by means of RO52 overexpression. Now, what does this Muscat assay system actually give an end result? So, the patients which show a higher level of expression of autoantibody, like giving a higher result in Muscat assay, those patients respond. It has been seen clinically that those patients show a higher level of, like if we use a treatment, specific treatment, we can cure the patient using this autoantibody, autoimmunity. But patients showing low level of signals in Muscat assay, they must opt for a different method of treatment. Now, the second, we actually worked on two projects. This was, I presented the first one, and the second part will be presented by him.
So, hello everyone. So, good afternoon to everyone, and also I'd like to present our, you can say this was a short supportive experiment that we performed. This was a molecular cloning of the gene PDCD1, which is basically a immune checkpoint gene. So, first let's understand what is PDCD1. So, PDCD1 basically stands for Programmed Cell Death Receptor 1, and it basically encodes a protein that is the PD1, and PD1 is essentially an immune checkpoint receptor, immune checkpoint regulator. So, it is generally present on, it is expressed on most of the immune cells, including the T cell, B cell, and specifically some kinds of natural killer cells. And so now the function of the PD1 protein that we expressed in the immune system. So basically, like every other receptor proteins, PD1 also has a ligand. So the two ligands that PD1 has are the PDL1 and the PDL2. When the PD1 protein binds to the PDL1 ligand, it basically leads to reduced T cell proliferation, reduced cytokine release, and also reduced cytotoxicity of any of the immune cells that are present in our body. So now how it is relevant to our work, because cancer cell, most of the cancer cells, basically certain kinds of the breast cancer and the pancreatic cancer cells, they use these misuse. You can say that they misuse this system to evade the immune system. So how do I do that? Now I would like to move over to the picture. So in the picture you can see the red cell, that is basically the tumor cell, and the green cell, that can be any kind of immune cell. Basically here I have showed it is a T cell, so but it can be B cell also, it can be NK cells also. So what it does, it is observed that any kind of tumor cell, they overexpress the PD-L1, which they were not supposed to express. So when they overexpress the PD-L1 receptor, and suppose a T cell that comes in contact or recognizes the tumor cell as a destructive cell, but since the PD-L1 receptor is PD-L1 ligand is present on the cancer cell, so there is an inhibitory signal, that inhibitory signal pathway that happens, and which leads to the reduced cytotoxic activity, reduced cytokine release, and essentially reduced tumor killing. So basically what they do is they use our own body systems, normal mechanism, to basically exhaust the immune system, and thus the tumor survives. And there are certain drugs like nivolumab. What they do is they interact with both PD-1 and PD-L1. They stop them from interacting with each other, and thus the immune system can essentially like recognize the tumor cell, and it can destroy the tumor cell. So now how PD-L1 and PD-1 system, this pathway can be useful in cancer diagnostic. Here we can see the doctor, when a tumor patient is admitted, so basically what the doctor does first, they test for a PD-L1 biomarker on the tumor cells. So if the PD-L1 biomarker test comes positive, like if there is a large number of PD-L1 ligand present on the tumor cell, that generally leads the doctor to a conclusion that it can be the patient can be cured with the immune therapy instead of other chemotherapy or other therapies that has a side effect, unlike normal immune therapy. So next we can see what is basically the PDCD1 gene and the PD-1 protein structure. So PDCD1 is a gene that is present on the human chromosome number 2 in the long arm, and it produces the PD-1 protein that is basically not a long protein. It is a 288 amino acid long protein, and it has 3 domains. The extracellular domain, it is the largest domain. It consists of 22 to 170 number of amino acids, and there are 2 other domains, the transmembrane domain and the intracellular domain. So basically the yellow part here, the ligand binding region, that is the extracellular domain. For our experiment, we only focused on this specific portion of the protein because it is the main part that is responsible for the ligand binding and other interaction with the PD-1 protein. So PD-L1 ligand. So basically what we did is we took this part and the specific gene for this part and used a vector to integrate it and perform the cloning. So this is the expression vector that we used. So basically we used a PBO1898 vector, which has a selective gene kanamycin R resistance, and also it has the 2 BAM-H1 sites. We can see there is the integrated gene is the PDCD1, that is our required gene, and there are 2 tags, like on the N terminal of the protein there is a his tag, and on the C terminal there is a seq tag. So basically if we talk about their function, the his tag is there for the ease of purification during later stages of purification, because his tag, as we know, it helps in nickel NTA column purification and also in the cobalt ion purification. And the seq tag, if we talk about, when expressed, the seq tag, it creates a molecule or expresses into a protein molecule that is a seq peptide. So what it does is basically it guides the protein after it is expressed to the extracellular region, so that it is easier for us to collect the protein after our experiment without essentially lysing the cells. And now if we talk about the experimental workflow, what we first did is the PDCD1, the vector clipping, and we used BAM-H1 high frequency to first linearize the vector. Then we did the infusion cloning. We put in a reaction mixture, we mixed the PDCD1 gene fragment and the linearized vector. and then it was transformed into DH5 alpha cells using heat shock mediated transformation, and then the cells were incubated for a short time for their growth, and then they were put into LB plus kanamycin plates for selection for the cells colonies that only the colonies that had the plasmid in them. And then we took some of the colonies and ran them in colony PCR using T7 primers. Sorry, there is a mistake. I wrote actually 17 instead of T7. So it would be T7 PCR using the T7 primers. So after this, we can get results of only those cells which have a successful cloning of PDCD1 gene, and then we ran the agarose gel electrophoresis along with the markers, which gave us the result of the positive colonies that had the PDCD1 cloned, that had the plasmid that is cloned with PDCD1. Then if we talk, then we took the positive colonies and put them in a liquid medium overnight for their culture expansion, and then what we did is we just extracted the plasmid, and it was sent for plasmid DNA sequencing. So what are the applications of recombinant PD-1 if we talk about? In our lab, it was mainly done because of immunotherapy research, cancer immunotherapy research, but it can be also used for producing designing new drugs or new antibodies that can be help in this immune therapy process. It can be used to prepare other drugs like nivolumab that is already available in the market. And lastly, I'd like to thank everyone, especially Sister Nivedita University for giving us this great opportunity. And also I'd like to thank Professor Junichiro Futami and also our lab mates. Without them, this project was it couldn't be completed. And lastly, thank you all. Now we'll take some questions, if any.
Any questions?
Hello? Yeah. So you purified PD1, right?
Yeah, purified PD1.
Right?
No.
Or PDC1.
PDCD1, yes.
Right. So how did you purify it? It came as a soluble format, or you had to go for
So basically what we did, it was actually impossible to do all the work within 2 weeks. So what our professor did, he already prepared the gene, the PDCD1 fragment. Right. It was readily available there. We just performed the cloning and the rest of the stages. We didn't prepare the PDCD1 fragment, but it was already prepared. It was ordered from some professional labs with the sequence. We got the sequence actually, but we didn't prepare the PDCD1 fragment by ourselves.
But they purified it?
Yes, they purified. Yes, they, in their lab, they purified it and they produced it. They kept it for their use.
Okay. But you're not aware actually, how they did purification?
No, actually, this was not the part of our project, work there.
No, fair enough, yeah. Anyway, thanks for the work actually.
Thank you.
Now, I have a question. So after colony PCR, you didn't send the clones for sequencing?
No, after colony PCR, we first ran the agarose gel. So
the gel, but after selecting the colonies,
yes, we sent for
the sequencing,
yes.
right? Otherwise, how you will know that? Because directly you said that after selection, you directly go for this plasmid mini prep.
Yeah, actually, I missed maybe, but yes, we sent for the Sanger sequencing.
Sanger sequencing.
Yes. Sir.
So after that, any other did you transfect the plasmid or any other further studies you have done with after?
No, ma'am. No, we didn't have enough time actually. And also we didn't actually saw the results also because our time was already over. We came back and then we heard from our lab mates that the result was positive. The Sanger sequencing result came and it was positive. So yeah, we didn't even see the result. So yeah, it was not enough time for completing all this experiment, because me and Shakhar, the total presentation, it comprises of 3 experiments. So it was the supportive experiment. The main focus was on the RO52 production. It was also going on alongside, but yeah, 2 weeks, and also not every day we were able to visit the lab. So due to because of the weekends also, so it was not enough time for us to complete. But yeah, it was a beautiful experience. Thank you.
Question?
Yeah, nice presentation. I just have a question from this slide only. Okay. How did you optimize this 15 base pair? You all did the optimization?
No, ma'am. Actually, everything that was required for the PDCD1 optimization, the fragment optimization, it was previously done, because this work is already going on for in their lab for years. We just went and participated in the work, but it was not from scratch. We did the work from scratch. It was already going on there, so everything was optimized, and we just performed this cloning.
All right, thank you.
Thank you, Shakhar. Thank you, Ritam. So we move on to our next speakers. The title of the next presentation is Exploring Gene Regulation, Cellular Differentiation, and Immune Response, a multi-project study. Will be presented by Piyasha Mukherjee and Prantika Maiti.
A very good afternoon to one and all present here, respected professors, friends, and to all my juniors. Today I, Prantika Maiti, and my friend Piyasha Mukherjee, we will be presenting our internship project, which we did in the Okayama University in Japan under the Sakura Science Student Exchange Program. The topic of our internship project was Exploring Gene Regulation, Cellular Differentiation, and Immune Response, a multi-project study which consists of 3 different independent projects. So this is the overview of the 3 projects. Number 1 is the CRISPR plasmid construction, which we did to check the gene regulation. The second project was on fusion index validation. It was done by performing 2 types of staining methods. And the third project was to check the metal ion effect on the inflammatory response on human immune cells. I'll be carrying forward with the project 1, and my friend Piyasha will be then presenting project 2 and project 3. So the project 1, construction of the CRISPR-I, which is interference and CRISPR activation plasmids. So the main aim of the project was to construct this CRISPR-I and CRISPR-A plasmids for the regulation of a certain protein, which we named as protein X. Different kinds of methods was used. First, we did the gRNA design. Then we did the cloning of the plasmid background. Then we did the validation by the gel electrophoresis, and also we did the Sanger sequencing confirmation. And mainly the main motto was to modulate the target gene expression without changing any type of genomic sequence. That's why we used here the dCas9 instead of only Cas9. These are the few terminologies. CRISPR-A, CRISPR-I, we use the guide RNA, which is the gRNA, and PAM. These were used for the target recognition sites. We used ChopChop tool that was used for the gRNA designing. And some of the few other methods like aniline transformation, restriction enzymes, we did MediPrep, MiniPrep, and lastly, the Ape gene software was used. It was used for the sequencing validation. Coming to the methodology, the first was we use this chop. This is a tool. Here we designed our a specific gRNA to ensure its specificity to our target protein X. And there were different selection criteria, like the target region was there, PAM availability, then like the efficiency and the other off-target considerations. And this oligonucleotides, the oligos that we have designed via this chop tool, these were ordered from the IDT, that is idtdna.com. Next, we performed the annealing. We mixed the oligo pairs and it was heated and slowly it was cooled down to form the double-stranded DNA insert. And two kinds of vectors were used. Like for CRISPR-I vector, like for the interference, we used the 1695 lenticrab. It is using the nucleus inactivated dCas9. It was fused with the crab repressor to downregulate the gene expression. Whereas in case of CRISPR-A activation vector, we used the AdGene 1719 lenticem, which was also the nucleus inactivated dCas9. It was fused with the transcriptional activators to upregulate the gene expression. Here also multiple sgRNAs were made to ensure reliable modulation. As you can see, for activation vector, we used samples A1, A2, A3, and A4. And for interference, similarly, I1, I2, I3, I4. So 4 samples for each was made. These are the vectors for activation vector and the interference. We took these images from the ApeGene software. Next, coming to the next methodology part, we did the transformation, like the plasmid DNA were introduced into the competent E. coli cells, and then we got successfully transformed cells by growing them into the LB agar consisting the ampicillin, and we got our successfully transformed cells. After transformation, we did the media preparation. In media preparation, we made a liquid culture of the LB broth containing the ampicillin, and also we made another replica plate. We picked up the single colonies and we inoculated them in the liquid culture along with the replica plate, and it was incubated. After the incubation, we did the mini prep. We isolated the plasmid DNA by performing the mini prep, and small aliquots were taken, and in that small aliquots, we added the loading buffer. Also, the enzymes like BAM-H1, NDEL, and BSM-B1, they were also used to ensure the cut in the DNA for further verification. Next, we performed the gel electrophoresis. Here in the gel itself, we also made 2 separate lanes to put our vectors there, like the 1695 interference vector was also added, and the 1719 activation vector was also added. And the gel was run. And for the verification, by the help of this AppGene software, we got the verification reference band of both the vectors, so that we could compare it and observe the certain band patterns and other changes. This is our result, and after using it, after comparing with the reference band, here we could conclude that all the samples from A1 to A4 and I1 to I4, they showed bands at the expected region, and they indicated successful transformation. So after the gel electrophoresis was done, we sent out our samples for sequencing. It was sent to the Microgen Japan for Sanger sequencing. And specifically, our sequence was focused on the BSM-B1 cloning site. So in BSM-B1 cloning site, the activation vector, it was expected a change at around CACC to GTTT region at the position of 2890 to 2900 kb, and for interference it was around 7790 to 9670 kb, along with this CACC to GTT region. When the samples were sent for sequencing, in the meantime, we did the MIDI pre for our confirmed clones and for further use in future, and it was stored. So this was the results. When the sequencing results came, we analyzed it with the ApeGene software, and we could see that in case of sample A1, we didn't get any kind of CACC to GTT region. But in case of A2, sample A3, and A4, we got the CACC to GTT region at the specific sites. Similarly, for our interference part, in I1, I2, we also got the region of CACC to GTT, and similarly for I3 and I4 also, we got the regions. So we could conclude from that that out of our 8 samples, 7 of the constructs were successfully, like the plasmid construction was successful. Only for A1, it didn't work, maybe due to, for A1, the gel band result was very much perfect, but in case when it was sent for sequencing, it was incorrect, maybe for some unstable insert or something else. We again performed the Sanger sequencing for A1, but the next time also the result was almost the same. So for the last thing, we did the validation part. This we couldn't perform due to the time shortage. So a validation was made that for protein X, it was identified through the bioinformatic analysis that it is a very potential regulator of C2-C12 myogenic differentiation. So we could have done some transcription level validation where we could have measured the mRNA expression of protein X using qRT-PCR and to check its regulation. And also some protein level confirmation could also have been done using Western blot or immunofluorescence to ensure that the mRNA modulation, it translates to protein level changes. And lastly, for functional validation, we could have used some myogenic markers like MYOD, myogenin, MHC, and to check the cellular differentiation. So our expected outcome should have been that for CRISPR activation mediated plasmid, there should have been an increase of the protein X expression, and it could have been enhanced the myogenic differentiation. And for CRISPR interference, it was expected to reduce the protein X expression and impair the differentiation. So these are the references, and the next part will be carried forward by Piyasha.
Thank you, Prantika. So I will start from project 2, in which we validated the fusion index using 2 different staining methods, cell mass staining and MF20 staining. So for this experiment, our model cells were the C2-C12 cell line. Cell mass staining uses the stain, which is a lipophilic membrane dye. So it intercalates in the plasma lipid bilayer and does not target a specific protein. Hence, irrespective of the stage of differentiation, we can figure out the membrane morphology and the cell boundary, whereas in MF20 staining, it targets the sarcomeric myosin heavy chain, which is generally found in the differentiated mature cells and not in the undifferentiated cells. So for MF20, we will get a better fusion index as the stage of differentiation progresses. These are some of the important terminologies. This was the methodology, just a brief overview. Now we come to the results. So as we can see, the results are not very prominent and there are some blotches. This might have been because the dye targets some lipids which were not present in the C2-C12 cell line, and hence the staining was not very prominent. It may also be due to several other reasons like problems in blocking. But even after we did some troubleshoot, still the results were same. So most probably the cell mass staining does not work for this cell line. This is how the result should come. This side is the proper result as it should come, and beside is our results. So we can see that this experiment was not that successful. Next we come to the MF20 staining. We can see that we are getting the myotubes. Now myotubes are the cells which have 2 or more nuclei, which is generally used to find out the fusion index. So this is the result for 24 hours. This is for 48 hours. Then 96 hours, 120 hours. So we see that as the differentiation progresses, our fusion index came better and we could visualize the cells better. So for C2, C12 cell line, the MF20 staining worked. So these are just some of the references. Now we come to project 3, in which we investigate the effect of metal ions on inflammation in THP1 cells for improved allergen detection. So we already know that zinc ions can induce the interleukin-1 beta secretion via inflammasome formation. And in this experiment, we wanted to see if some other metal ions like calcium, magnesium were also showing the same properties. So we used 2 assays to check the cell cytotoxicity and the interleukin-1 beta secretion. I will come to it. So these are some of the important terminologies. Next we come to the cell titer glow. So once we did our experiment, we cultured the cells, we stimulated them using LPS and the metal ion treatment. Then we separated the supernatant and the cell pellet. So in this experiment, we used the cell pellet to check the cell cytotoxicity. So we have the luciferin enzyme which reacts with the ATP, which is present only in the live cells. So only if the cells are alive, this will show results. Hence, as the light, as we get more signals, we will see, we can understand the lesser cytotoxicity. So next we come to the LUMIT assay. This is the assay for checking the interleukin-1 beta secretion. Basically, in this experiment, we have the luciferin, which has 2 fragments, nanoleuc fragment 1 and nanoleuc fragment B. So both these fragments will bind to interleukin-1 beta at 2 separate sites, and then it restores its enzymatic activity, after which it will react with ATP to produce light, and we can visualize it under fluorescence plate reader. So the light signal is proportional to the interleukin-1 beta concentration. Now coming to the results, 11A to 11H we have our standard and the rest were our results. So as we can see, the cell cytotoxicity was very less, which is a good sign that the cells did not die. Now coming to the LUMIT assay, there was some problems. 11B to 11H and 12B to 12H we have the standard, whereas 1D and 1H were our positive controls. Now we can see, compared to the standards, the interleukin-1 beta secretion was very less. So it might be that the LPS stimulation did not work properly, or there were very less cells than were required. So this was not that successful. These are just some of the references. Now, we also wanted to learn cell culturing, and we requested our professor. So we performed the cell reseeding on B16F10 cell line, and this is what we did. So I would like to thank everyone who made us go to Japan, for which this Sakura program was a success, and also to our supervisor, Dr. Ayano Sato, and our lab mates. And thank you to the university, to everyone involved in this program. Thank you.
Any questions?
Thank you. I just wanted to know that what is your experience about the lab culture in the Okayama University? Because you people have spent more than 3 weeks like that. So you must have experienced the lab culture there. So what is the experience in terms of the lab culture there?
So the lab culture there is not that different. What is different, I would say, is the instrumentation. They prefer up-to-date techniques which will take very less time, and hence we could perform so much in just 16 days. So I would say that is the point of difference for which they are so advanced. It's not that the lab culture is greatly different. Mostly the techniques that they use, how up-to-date they are with today's techniques, that is what is the point of difference between both our labs.
Only the instrument is the only thing.
If we go to labs, even in SNU, I have the similar experiences, good professors, I have had a very good mentor, I have learnt a lot of things even in SNU. So I cannot discredit our university entirely. But what I found different was that they were very much advanced in terms of use of technology, in terms of interconnecting dry lab and wet lab, which I would say is the point of difference again, because I used to be very bad at dry lab, but there I started growing interest in dry lab. So it was same for both of us actually. We were very bad at that, but we learned a lot of things there.
Thank you.
I think Madam was asking about the lab culture, means the work culture over there, like how people do work here and how people do work there.
Again, I would say, sir, there is not much difference because we used to go there at around 10:30 in the morning, and by 5 we would be back. And even our professor used to say, "When are you going for lunch?"
We used to do it at around 12:30. Our supervisor, she used to tell us, just go and have your lunch. Then you can come back in the lab and continue with your experiments.
That's good for internship students. Okay. Great. Be a PhD student and then we will see. Okay, anyway, so those immunofluorescence pictures were confocal or immunofluorescence microscopy? Those pictures that you showed in the slides. Yeah.
Immunofluorescence microscope.
So you did it yourself?
No, our lab mate helped us. We don't know how to operate it actually.
You don't know how to operate it. That's why if you had known to operate it, you could have got better pictures. Anyway, thank you.
Thank you.
Thank you, Prantika. Thank you, Priyasha. So now I would request our the last speaker, Srija Shah. She will be presenting on her project, which is entitled Characterization of Hydroxypeptide for Biomedical Application. Sorry, hydroxy? Oh, but here it is peptide. Sorry, it's appetite.
Hello, everyone. Good afternoon to the professors and all the students present here. I'm Srija Shah. Today I'm going to present on characterization of hydroxyapatite, which was my work in Japan under the supervision of Professor Satoshi. Okay. Before I start with my experiment and everything, I would like to give you a brief discussion about what is hydroxyapatite. This is an inorganic molecule which was important for the formation of bone and teeth in our body, and it has a good biocompatibility and osteoconductivity. So the aim of my experiment was very clear. First, I need to synthesize the appetite. Then I go for the characterization of the appetites. So for my own sake, I distributed my own work into 3 different parts. My first work is to know how to synthesize these appetites, then to characterize the appetites, then the results. So first, as a raw material, I have alpha TCP powder, from which I need to form the hydroxyapatite. So first I go for the hydrothermal synthesis procedure. For that, I put 1 gram of alpha TCP powder in a taphenol flask with 50 ml of distilled water. Then I put it in a drying condition. I do my experiment in 2 different temperatures. One is 80 degree and other one is 150 degree centigrade. Then I put it for the incubation around 24 to 48 hours. But as we have a time shortage, so I did it for 24 hours. Then on the next day, I go for the XRD analysis, which was known as the X-ray diffraction. So I did it because I want to know my appetite is formed or not. Then I go for the FTIR analysis. Through FTIR analysis, I get know about the chemical bonds formed in my appetites. And I would like to add one thing in it. As I said, hydroxyapatite is very good for, is very important for our bone and teeth. And this is, if the hydroxyapatite is calcium deficient, then it's very useful for the tissue engineering. Next. Then after the FTIR, I go for the same electron microscopy. Through this, I get know about the structure of my formed appetites. Then I go for the bed analysis. Through bed, I get know about the surface area of my appetites. Then I go for the TGDTA, or thermogravimetric differential thermoanalysis test. Through this, I get know what is the effect of the temperature on my formed appetite. Now come to the results. This, as we can see, this is a XRD result graph. The first one is product appetite, which they already formed. Next two are my formed appetite, 150 degree centigrade appetite and 80 degree centigrade appetite. And the lower ones are the available appetite PDF data and alpha TCP PDF data. Through this, we know that there is no similarity between the alpha TCP PDF data with my formed appetite, but there are similarity in the peaks of the appetite PDF data and my formed appetite PDF appetite data. So through this XRD process, I get know that there are hydroxyapatite formed in my samples, but there is no presence of alpha TCP. Then FTIR graph. This is the FTIR graph. Through this, I get know that in my appetite there is hydroxyl group present and also phosphate group present, but there is a flaw in my work that I found out there is a CO3 formation also happened in my appetite. So I consider it as a flaw of my work. Then this is the same pictures. This all are in the 2000 magnification. Through this picture, as we can see, the structure of the appetites are needle-like crystal structures. These structures are very good for the biomedical tissue engineering purpose. Then this is the BET analysis result graph. The product appetite has the surface area around 42, but my produced appetites has 15 to 16 meter square surface area, which was actually happened because I think my produce appetite from stay as a crystal form. So the absorption around the molecules are get converted, and I don't get the proper data. Then this is the thermal analysis graph. As we can see, the first first lower one peak around 100 to 200 degrees centigrade. That refers to the water loss of my components. Then again we have a lower peak in around 839 degree, which was indicating about the carbonate decomposition, as we find out in the FTIR result also, that there is a presence of carbonate group in my samples. So, as a conclusion, I can say that I like to say that I successfully produce hydroxyapatite, but there are some flaws in my apatites. As a flaws, which I can say that in the higher temperature, maybe my samples convert into beta TCP in the place of hydroxyapatite. That is why, that is what reason I found a carbonate group also in my sample. And as a future perspective of this experiment, I can say that if we can use nanostructure or advanced level ion substitution technologies, then we can use this type of thing for the tissue engineering or bone regeneration, which was their present work in Japan. Actually, I don't know about details in it. At last, I would like to thank my professor there, Dr. Hawakawa Satoshi, for his help. Also my lab mates present there. And last but not the least, SNU for this wonderful opportunity. Thank you, everyone. Thank you for your time.
Any questions?
Okay, so in a microscopic image, you claim that the needle are better for biomedical application. Why is that?
Biomedical application, I don't know very detail about it because they are going through this process, but how much I know, I can tell you about that. They use this product as a material, as a bone filler in their experiment. The present work they are doing right now, that they are using hydroxyapatite as a bone filler. And as much I know, and the professor said me that this structure, this raft structure or something, this is very important to work as a filler in the bones. But the commercially produced apatite actually has a smooth surface, which is not good for the bone fillers. So we actually don't able to use that type of appetites for our bone fillers. But my experiment appetites has the rough surface, so we can use that. More questions?
So it's on. Okay, so you have done same. So
yeah.
What do you do exactly?
On the first day, my who was helping me, he did all the things. But on the next day, he allowed me to do that. So I did the same myself.
Yeah, what you did?
Okay,
Steps.
so first I placed the sample on the plate. Then I put it for the coating purpose. I put it in for the platinum coating. Then I kept out around after 30 minutes. Then I put it in the machine. Then we operate in the computer.
It was operated by the technician. Okay.
We get the picture.
Okay, thank you.
Thank you.
Thank you so much, Srija. Srija.
Thank you.
Thank you.
Thank you.
Thank you, all of you, all the students. You have wonderfully presented your work. So now I would request our present HOD, HOD of Biochemistry, Biotechnology, Dr. Sudipto Shaha, and our to please and obviously, and our dean of our school to come upon stage and felicitate these students. Sir, so I would request our Pro-Chancellor, sir, to also come upon stage, and please, they would love to get felicitation from you. So they would love to get felicitation from you, sir. So now I would students, Swapnil Pal, Swapno Neil. Sorry. Shakhar Shom. Ritam Pal. Prantika Maiti. Piyasha Mukherjee. And Srija Shah. Students, please come for a group photo. And I would, yes, Shrujit, AB, Anindita ma'am, Amarnath sir, Siddhartha sir, Iman ma'am. Sir, please, Dr. Shobhashaji Haldar, sir, please come. Yeah, come. Thank you. Thank you, everyone. Now we move on to our next session. In this session, I would request Professor Dr. Amarnath Mukherjee, Assistant Dean of School of Life Agriculture and Biotechnological Sciences, to come upon stage and introduce our guest speaker from industry, Dr. Shobhashaji Haldar.
Thank you, Sani. Respected Pro-Chancellor, sir, respected Dean, my colleagues and students. It's my pleasure to introduce our distinguished speaker today, Dr. Shobhashaji Haldar, a senior biopharmaceutical professional with over 2 decades of combined experience, which spans across both industry and academia. And he has a deep expertise in CMC, which is the chemistry, manufacturing, and control of biologics and biosimilars, and also analytical method development. And for someone who has worked in industry for a while, we in academia sometimes forget the importance of CMC or the manufacturing in the industry, because whatever product we are using, we do the discovery part, but ultimately it goes to the manufacturing, and there actually brings the product into the market. So whatever drugs you are taking, whatever pharmaceutical product you are using, there has been a huge amount of contribution from the people in the industry, R&D, in the industry, and who works in the regulatory submission or in the CMC control department. And Dr. Haldar is one of them who has a wealth of knowledge in the field of biopharma development. He earned his PhD from protein science from Ross Institute in Calcutta, followed by over 6 years of postdoctoral research in USA. He then went on to contribute more than a decade in leading biopharmaceutical industry, which includes Pfizer in Chennai, Pfizer Biologics in Chennai, Syngene International in Bangalore, and Origin Pharmaceuticals in Hyderabad. Throughout his career, he has led multiple research projects, contributed to numerous peer-reviewed international publications, and played key leadership roles in biologics regulatory submission and product lifecycle management. So currently, he is engaged in consulting for CMC analytical development and has been instrumental in setting up various CRO and CDMOs. So please join me to warmly welcome our speaker, Dr. Sabyasachi Haldar.
I need a system. Hello. I need also this one actually.
Okay. It's on.
It's on. And what is the pointer actually?
Just give me a minute.
Sure. Anyway, I can start actually, yeah. Saiful, students are I need a control actually. लैपटॉप को कनेक्ट करा जाएगा ना
ऐसे लैपटॉप कनेक्ट करा जाएगा
করা যাবে তাহলে তার তো ল্যাপটপটা দিলেই হবে তো Sure. Well, yeah, thanks. Can I start, right? Yeah. So thanks, Dr. Amarnath, for a nice introduction. I didn't realize actually, this, if somebody can be explained actually, my expertise in this way actually, the slide actually, whatever you guys made. So I would like to start from the kind of respected Dr. Dhrubajyoti Chattopadhyay. So I personally did not have a chance to kind of the part of his the classes in the university also. I did my master's for the chemistry actually, and then I think during the PhD time, sir, you came for some time actually, for some review work, but for during my my presentation, I did not have kind of chance to show you actually. But anyway, and then all the faculty members in SNU and students here. I'm really kind of privileged and honored actually to be part of this kind of talk in this today's whatever the students presented. Actually, the 767 students actually presented their work in the 2-3 weeks, whatever they spent in Japan. So anyway, yeah. So I would like to start today's presentation. Is a why I said actually a workshop. There is some kind of is it not working? Or can I use this one? Yeah. Yeah, so for the workshop, actually, it's always require kind of whatever Dr. Amanat was asking about the infrastructure. So infrastructure is always good to have to make an effective workshop. Kind of the lab as well as the theory, practical, everything. The combination if we make it. But the idea was to kind of hear majorly the introduction and the induction about the biologics and the biosimilar. What's going on in the biopharma industry. So that's the main idea. I took 1.5 an hour actually for time. It's quite a kind of long slides. I'm sure I see a lot of students already left. A lot of students already talking each other. I see actually. So guys, please actually kind of a small request for you guys, students. Just kind of focus on in a few minutes or half an hour or 1 hour time I will take from you guys. It can be helpful for you guys actually. So today's the main purpose for this conducting workshop. So I am personally I came from an academic background. I did my PhD in Bosch Institute in protein science, and then went for a postdoc, continued in the same area actually, and then also I can consider as my independent career in research in industry actually is the same almost area. Scientifically, there is no such difference in industry and academics, but absolutely, operation part as well as the presentation part and the representation of the science, how to get the molecule approval. So I'll just give you some brief understanding today, and let me see actually, how much I can cover today to reach to you guys. So the idea actually, to not to the present only. So I would also, kind of understand how much you guys are, listening and, kind of to interact with me also. So I, kind of wanted to know how much you guys are interested about to biopharma industry, as well as how much you know also. Like, it's nothing wrong to, kind of if you don't know anything. In fact, I'm also still learning every day actually. So I think the next slide is not required at all. Dr. Amarnath Mukherjee already, kind of covered everything, whatever I did. So what I was just trying to say in here that the Pfizer as well as Origin and the everywhere I did a lot of science, but the presentation of the science a little bit of different way actually, but technically there's no difference. So that I'm going to discuss next 1 hour. So today's workshop, this is the summary actually. So the fundamental of biologics and biosimilar. So then I'll start from the discovery to how to, the development and developability. And then the regulatory guidelines and the opportunities, challenges, and future. That's the title of the workshop. And actually, I kept some of the quiz session. It's just very simple questions. Don't worry about too much. It's just kind of keep you guys' attention to kind of to and scope to interact more actually. And finally, I just like to introduce about the new initiation from my end, and that's a Biovigyana Biopharma actually. It's not, it's from it actually. It's very early stage, but I thought, kind of it's a good platform to present here. So let's come to the first topic actually. What is the biologics and biosimilar? I'm sure actually you guys all learned and understand about what is biologics and the biosimilar in academics or in whatever Dr. Jesus was saying actually, like this COVID time we talk about a lot of biologics, lot of biosimilar, and a lot of funding, lot of improvement. What can be utilized actually the bio molecules as a therapeutics actually. So far we whatever go to the pharmacy counter you get all a small molecule, ibuprofen or Dolo 650, paracetamol, everything. But but the use of biologics and biosimilars so far is very less. It's quite expensive. And also it's not, always used for the regular use actually. It's we only use for the critical diseases. But one of the biologics, before it was not a part of the biologics, right now we included insulin actually. So I'm sure the insulin, right now, almost most of the families, I see that at least one of the people, they are, diabetic patient. So either they use the insulin. So insulin, the pathway, before it was considered as a peptide, right now the insulin also considered as a biologics. Now let me, go to the what is the biologics. So biologics are the therapeutics products that derived from the living organism. I'm sure actually that everybody knows about this. Whatever I'm just talking about, the vaccines, the blood components, monoclonal antibody, gene therapy, cellular therapy. So you guys do a lot of, the research on these topics as well as this kind of the molecules. You can be, heard about that this is the molecules, a blockbuster molecule for the cancer therapy as well as some rare disease actually. So this is all are biologics. The biologics majorly they derived from the living organism. So next, the characteristics of the biology is that produced in the living cells is a large and complex molecular structure, and then the manufacturing process and storage condition is very sensitive because it's a protein. I'm sure the protein storage is always a little bit of challenge rather than to storage of small molecules, and it requires a strict quality control and cold chain actually where the storage. So now I'm just coming. I'm sure the students and the faculty member also kind of knows about whatever the picture I'm showing here right now is the antibody, typical antibody structure, the top left corner actually here. And here this two part actually it has a IgG1 has a two part. One is a Fc domain, another is a Fab domain. So this is the domain. Can I get a laser pointer? You have it, or I can use my hand actually. Okay, not an issue. So this is actually the area actually is called the Fab region, and this is the Fc region, and sorry, the other way, Fc and Fab. So this is actually the receptor binding domain, and this is the bispecific antibody. So bispecific antibody, so here the antigen binds. I'm sure people know that actually here. But the bispecific antibody, so this is what I can say is the current. The MAPS is quite established last 10 years, people are working on that. So new, what I can say the advanced biologics is the bispecific antibody and the multispecific, will come together. So bispecific antibody, what happens? Like the one arm, if you see that, it can bind to the one antigen, the other arm actually it can bind to the other antigen. So once we inject this molecule, so it can show the two actually the action all together. That's the advantage for the bispecific antibody. And in that way, so I think one of the students also explaining about the PDL1. So, the industry, I cannot say the name of the industry. There's a lot of, limitation about to show the data from the industry. I'll show today overall, the operation part, the biopharma industry, and majorly, kind of the knowledge and understanding actually. So this PDL1 and the fusion, actually what happened, like this antibody, what we do, sometime what we use actually here, this is all the arms. We take a different protein actually here, actually here. So that's called fusion protein, and that's a multi-specific protein. So what can happen? So here is the one hand, another hand. We can use actually the two other side actually for the different antigen binding site. So in that way, so one molecule can helps to the multiple functions. So if you see, and there are a few other formats also. So if you just consider, this is a heavy chain. It's called a heavy chain. This is a one actually single chain, and this is a light chain. So if you see actually here the different format of the multispecific. So where you can see actually, so this domain we just consider sometimes this domain, sometimes consider this domain, sometimes we consider only the heavy chain, sometimes we consider only the light chain. It depends upon the where we are going to use this kind of all the chains for the different function. There's another, the class of the molecules. in industry. It's very early stage, what I can say in India actually, but not in world, the ADC. It's a quite challenging, but what is ADC? ADC actually what the small molecule we attach to the antibody. So that's called an antibody drug conjugate. And there are some few already the approved molecules in the market. But right now, the entire, I can say, including the world actually, they are focusing on the bispecific ADC and the multispecific ADC. So what is the advantage actually? We know the small molecule we use for the different disease. If you use actually the antibody, it is always easier because some of the small molecule have a toxic effect actually. So once you attach to the antibody, so the toxicity will be reduced. So that's a helpful kind of advantage to use the ADC. But the problem actually the ADC is quite a challenging to make. Because why is the challenging? Because ADC, the antibody and small molecule, there is another we use actually cross-linker. So the cross-linker, the choice of right cross-linker and how it operates inside the body is always challenging. So people are doing a lot of research. I see that recently also, kind of government of India and the DVT is also funding on this, doing the research on for the cross-linker. So this is very, very much early stage, what I can say for the bispecific ADC, but some of the ADC, only the antibody is already established in the molecule, in the market actually. And also the other format also. So other format is also not established, is very, not I can say the early stage, but few molecules are in the early stage. Few molecules we are in the different expression system because generally the mAb we express in the mammalian cell, but some of the cases also we are using the E. coli cells to reduce the costing, as well as the E. coli is always easy to handle. So that's overall, I can say whatever we are using in biopharma industry in India. I mean, absolutely, there are a lot of research is going on in the entire world. I'm not going to cover, but this is almost this kind of the molecules we are targeting as a biological biotherapeutic medicine. So in this treatment, whatever I discussed actually, it's this biologics generally we don't treat for the regular disease. So this disease like kind of rare disease like the cell and gene therapy, like the CAR T cell therapy, it's all are using. It's quite expensive actually, and it's a very rare disease we use actually. And then cancer, diabetes, and then majorly the immuno-mediated other diseases also. So there we can use this all the biologics molecule. It's quite expensive, but enter the effort overall industry is to reduce the costing, to how to reduce the cost so that it can reach to the general people actually. So whatever this is few molecules, the biologics molecule, it's adalimumab, rituximab, and I'm just wanted to give a understanding about this. All are monoclonal antibody. The most of the monoclonal antibody is already in the market, and if you see this is all a big organization, AbbVie, Genentech, Amgen, and Eli Lilly, already they already kind of what I can say is shipped to the market. It's already available in the market. And few therapeutic proteins are there, like whatever I can say is the insulin. Insulin is quite we are using for the regular basis. Filgrastim is also using actually whatever the chemotherapeutic agent we use actually. They are a mixture of the smaller molecule and there is also we use the filgrastim actually. And this is the treatment actually. So just summarizing everything, all the information in one places. But wanted to mention here actually whatever you see so far the available molecule is a molecular antibody. Not the bispecific as well as the ADC is not too much available actually. So majority of the market is still the monoclonal antibody. The research is started long before and already a lot of molecules already in the market. In fact, India is also kind of launched few molecules already. So I think now I'm going to introduce what is the biosimilar. So I think this biosimilar part we are actually what I can say the Indian biopharma industry is enriched. The biologics research, yeah, absolutely we are trying but that's require a lot of funding, a lot of time also because one biologics drug it takes almost 10 to 15 years to build up. But biosimilar is just a similar, is a copy of the biologics. So when the biologics everything is in market, it has some patent. Some of the date of the expiry of the patent. So after that, any organization they can launch the biosimilar. So right now, last I think 2019 it started actually. All the biologics, this 10 years or 15 years of patent is expired, and that this last month, I think in March, there is a one blockbuster molecule, the semaglutide. So that's using for the weight loss as well as diabetics. So right now, all the companies are launching. If you see the last, if you follow the all news, pharma news, almost I can say all the big companies, including Biocon, also launched this semaglutide actually. So that's a challenge. So biosimilar company they plan for at least the 5 years before the patent expiry, and what they do, they just make a same copy. What is the purpose? To reduce the costing. Main purpose to reduce the costing. But biologics drugs is very expensive. So once I can say as a definition, this is a copy of the biologics medicine that is similar but not the identical. It's a really challenge to make the identical molecules for the complex molecule like a protein. I have actually, in fact, insulin also there is a lot of challenge. Those are not big molecules. And it's a biosimilar medicine. Generally, the cost less than the original brand, the biological medicine, and they are just as safe and effective. So we have to ensure that whatever you are producing is a safe, it's a potent, and it's a less costing. Until unless if you don't make a less costing, it doesn't help too much. So most of the organization, whatever I see, the 20 to 30% it reduced, but R&D, the research work is people are working on this to reduce the costing of this biosimilar. So US FDA is a regulatory agency. I'll come to that portion. So US FDA, they defined actually the biosimilar. So the biological product is highly similar to the reference product. So reference product is innovator biologics molecule. And the product notwithstanding the minor difference in the clinically inactive component. So I think this is the the big catch in this line actually. So what is saying actually here? So once you purify protein, so we say the protein is more than 90% pure, or maybe 95% pure, or maybe 100% is not. We cannot actually say anything 100% pure. So the 90% molecule is similar to the original molecule, but here the challenge actually that the rest of the 10%, whatever the 10% the component, we have to characterize. And we have to see that 10% is not impacting to the anything in the clinical. So I'm just curious, like you guys are understanding actually the students are sitting quite a far actually. If any question, just stop me and ask actually. I'd be happy to answer if you have any question. This is a basic actually textbook knowledge. I'm trying to introduce you guys so that I'm sure you may actually heard about all the terminology, but I'm sure there is a few sentences I added here. So what I'm just trying to say, the protein once you make the protein or MAB purified, that's 98 generally 98, 97 or 99% purified. So the 1%. The impurities, whatever it has, in the protein, so that we have to ensure that it is not going to be impact in any immunogenic activity. So whatever the purpose of the molecule, we are making some copy, right? But some of the other, whatever the impurities, it should not impact to the patient. So that's actually the target. So it's not necessarily always you have to make it identical. Like the biologics is 98% or 99%, your biosimilar can be 97% purified. But you have to ensure that whatever the 3% is there. So that you have to show the character. So it's a very strict US FDA, expectation. So like academic background, I'm sure everybody published the paper. So during the publication paper, you submit to the different journals and then come for the lot of queries. The same thing US FDA, and this US FDA for the US actually, the EMA is for Europe, India is a CDSCO. So once you submit actually the molecules with some documentation, so they ask for a lot of questions, you have to answer. So this is the agency actually. They approve. After this approval, we can go for market the molecules. So now the biosimilar and the whatever the generic. So generic medicine is just generally the small molecule, and you can make an identical molecule. Generally we try to purify almost more than 99 or 100%. And it's we have to get an identical one, the generic versus the small molecule. And there are, a lot of progress also. I think quite India, this research, a lot of, long before we started of the generics molecule. So that's how we actually, we got a small molecule is a very less, in price. But in the biosimilar, like the all other biologics, is subject to the inherent minor variability. So there might be the minor variability is there from the biologics as well as the biosimilar. But the both biosimilar and generic offer from the potentially more affordable treatment option. So both the target is affordable. So generics is the copy version of the small molecule, and the biosimilar is the copy version of the biologics. So now this is the novel biologics and the biosimilar development. So once, if you focus and if you take a look on how the development, the entire development is in a different way. So what I actually wanted to show in here, So if you see actually here, this is the clinical studies, this is the clinical pharmacology, non-clinical, and this is analytical. So this is the entire content of the biologics development study. So once you go for a regulatory file. Once it comes to biosimilar, so additional clinical pharmacology, the non-clinical study, and analytical similarity. So if you just take a look, I just wanted to introduce here. If you take a look on the entire developmental pathway, the analytical for this novel biologics requirement is very less, and the clinical and non-clinical is very high. Whereas once you go to the biosimilar, so biosimilar, the analytical and similarity from the biologics as well as the biosimilar, this similarity, you have to make a good amount of the data so that you can convince to the regulatory agency that the biologics as well as the biosimilar, there is not significant any difference, and they are all clinically safe and potent. Next is the interchangeability in biosimilar. So interchangeability, the biosimilar is typically for the US, what they do actually. So if the biosimilar is a quite potent, very highly similar. So what generally the organization, the companies they do, so they actually during their clinical study, they exchange with the reference biologics. So they start with the clinical study with the reference biologics. Then in between they use actually the biosimilar, and then again the reference biologics. So it's a switching kind of. They do multiple times to ensure that whatever the biosimilar they are using, it's very highly similar to the reference biologics. So in that scenario, what they happen? So it generally the pharmacist in this actually it's already approved in the US, not in the India and other country actually. The pharmacist can give this interchangeable biosimilar. So only the agency, regulatory agency, they approve it can be used as the interchangeable. So like the doctor suggests one prescription, one drug actually, if you go to the counter, if they don't have actually, they can use actually the other interchangeable biosimilar. So that kind of the authority the pharmacists they have, and that's who gives actually the regulatory agency. So now why the biologics are dominating? Absolutely, so far if we consider the last 6-7 decades or more than that actually, the way actually the small molecule utilized to cure the disease, the biologics is still under development, I can say. But slowly, why the biologics are dominating in the pharmacy market actually? Last 10 years if I consider, is significantly hugely impacted overall the market in the biologics and biosimilar. So why? So number one actually the super target specificity. So what happened? Small molecule once we eat something, take some small molecule medicine, there is a lot of non-specific interaction happen that we call a side effect. So we ensure that what is the side effect is there, because that molecules along with the target, it also attacks different other organs also. Some of the cases small molecule can damage the other organ actually. That it can cure the target cell, or it can help to the target cell, but it can also kind of damage the other cells. That's why actually we see we can say actually that there is a lot of side effects we say. But the biologics drug, typically, it has a very typically, specifically bind to the antigen. So the advantage actually for the biologics is like the side effect will be less. You're getting my point actually, right? So that's a major, I think the major point for the biologics drug. And absolutely the higher efficacy, because we use a monoclonal antibody, whatever the proteins we are using is all are in the human body actually. We just, kind of scale up some of the cases like some of the proteins or some of the antibody. So next, actually, the breakthrough in the complex diseases like the cancer, wherever I see actually the monoclonal antibodies, some of the antibody like adalimumab and the rituximab, trastuzumab. Actually, this is actually they gave a really significant impact on the cancer different indication and the autoimmune disease. In fact, the rare genetic disease. The rare genetic disease generally today's cell and gene therapy, usually the gene therapy you are using. Cell therapy, I can say people are exploring things, but gene therapy is already in the market. It's still in the very expensive, but a lot of R&Ds, a lot of companies are working on to reduce the pricing actually. So that's actually kind of the main reason why the biologics predominating in the pharmaceutical market. So if you see this actually, the overall market in the biologics, so 2024, this is like 2 years before, 744 billion, and the way actually is increasing, it's a 2030 and to the 35, its target is 2.4, trillion. Biosimilar is also increasing the same way. If you take a look on the 2024, is a 35 billion, 40 billion, so almost I can say the 5% of the enter the biologics. Typically, the biologics are very expensive and biosimilars less. So that's how actually the costing. But overall, the biosimilar also taking the place. So, and India, India is actually the biosimilar hub. So most of the organization they produce the biosimilar rather than the biologics because of the kind of the huge costing, the R&D costing. Yeah. A lot of academic lab they already initiated for the biologics research, but industry it's still a little bit of behind. So hope you guys also know actually maybe kind of this Biopharma Shakti. There's a one new government initiation actually. So they almost approved 10,000 crore actually funding to just boosting the biopharmaceutical manufacturing and to make the biopharma readiness skill development from the students. They already funded, a lot of NIPAs actually. So I'm not sure of which NIPA, but so far that's listed actually. Because since it's a huge demand of resources actually, so in biopharma industry, and we are not getting actually what I can say actually, the people are not well skilled, and most of the cases what happens the freshers they don't have enough kind of in their syllabus there is not too much actually. Kind of whatever we do. I mean, it's always, some of the expectation from the industry that at least some of the basic knowledge. So once you understand something about the water biology, biosimilar cell and gene therapy vaccine, it can help. It can help to get a internship or next level of the once you explore to industry. Yeah. Absolutely, along with the academics, there is a lot in the industry also is the potential place from the masters. PhD. In fact, after postdoc also you can always to go there, and there is also all we do in the science actually over there. But the presentation absolutely that the agency the way overall is a little bit of different. So now if you see this the overall market, the mapping of the for the biologics biosimilar in the world. So Asia is almost like the around 15 to 18%. Absolutely, the North America is the mostly they started long before actually the R&D and the launching of this product, and the other countries also. So almost 40% global pharmaceutical revenue is the biologics and biosimilar, and it will be for sure the increasing because next 10 years you'll say actually more in this space actually. So now is the quiz session. I feel I'm just kind of to motivate to the students. And kind of few very small question I set off. I'm sure it's a very easy question. So I just want to listen from majorly from the students, right? Can you and whoever want to answer actually, can you just introduce yourself? Yeah, please. Option B. Anyone else actually would like to add, or all are option B only? Anybody want to say that something is different from the option B, or just this is a locked actually? Okay. Yeah, good. Yeah, thanks. Can you introduce actually look your Okay, thanks, sir. Next question is actually, which of the example of the biologic drug? Somebody from this side actually. Nothing wrong actually, yeah, please. If you are, correct or incorrect. Option? Option C. Anybody else actually want to add? Option D. So you are saying the C and you are saying D. So which of the following is an example of the biologic drug? So biologic drug, C is the monoclonal antibody, and next is the ibuprofen. Why you are feeling actually the D, and why you are feeling the C? Fine. Now ibuprofen is actually the small molecule, right? So it's the answer is a C. Next, actually, the back side. Somebody from this side actually.
Option C, the highly similar versions of approved biologics.
Right. Anybody else want to add anything other than C? It's final, right? Or no? Okay. Anyone again this side? This side, but back side, not the front side. Yes. Option B. Option B. Anybody else?
Option B. Small molecules.
So it's option B only, or anything else? So biosimilar differ from the generics because the biosimilar are chemically identical. No, right? Option A is no possibility. Biosimilar are produced from the living system. That's the option. Biosimilar are cheaper than the generics? It's not. Absolutely not. The biosimilar do not record the clinical trials. This is also not. So option B. Done. So this is the last question, 5 question. The term reference product refers to, again this side, back side. Option B. Everyone? Sam? Done. Thank you very much. At least, So, guys, actually, you guys, I don't know, actually, Surjit or Siddharth, can you add, actually, you guys teach this topic, actually, to the class, or Amarnath, can you add, actually? I mean, just to understand, actually, how much the students understand, or is all are new topics? And there is a really, all the 5 questions, everybody is answering, actually. Okay. Right. So, yeah, I mean, this part actually I'm going to cover. There are few more actually, section, but I'm sure it will be too late. But let us understand today actually how much, we can learn actually. So, but every section have a 4 or 5 questions actually, the quiz session. And it will be nice actually, all the students, listen and, participate this session actually. So here we are going to discuss about how the molecule. So far we understand what is the biologics and biosimilar. Here the discovery to development, how we develop the biologics molecule. So discovery, I'm not going to discuss in details about the discovery, but I'm just giving the brief understanding about the how we do actually the discovery. So first we do the target identification and the validation. So and then just to screening, understanding different the target actually different molecule. We check actually affinity specific because every molecule once we go for a screening we have a target actually some indication. So based on the indication, based on the specificity and the potency, we make a screening. And out of the screening, actually, you may get actually 30, 40, or 50 leads, actually, the lead molecules. So that 50 molecules initially, I mean, a lot of people also today introducing with the different plate and different cell line development here, right? I'm sure you guys also learning, actually. So that kind of, all the leads, what we do, we get the lead candidate. So out of 50, maybe we go for a 10 lead candidate. We don't go for a 50, actually, the best 10 candidate, actually. And then what we do, we just check the durability assay. So what is the durability? So durability, actually, every clone, we have an understanding that every clone has a, they'll express the protein. But once whatever our understanding, if you see actually the 10 clones, if you kind of grow actually, scale up, like from the plate to the flask to the bioreactor also. So if you see actually that every clone don't have a same expression level, same VCC actually, the same survival rate actually. So based on that actually, we check actually the 3-4 molecules. And other than that actually, so what we check actually the scalability. So in R&D lab, all the research lab, in fact, during my academic career also, whatever I was used to purify protein is a milligram level. But whatever the industry we purify as a kilogram level, some of the cases gram and the kilogram level. So we ensure that all the proteins, whatever we purify, whatever we discovered at the early stage, that molecule can be scaled up to the kilogram level. So that's some preliminary assay, because otherwise what happened, so one CMC development, it takes a few years, after 6-7 years. If you see that this molecule is not working, again you have to go back and start the activity from the different leads. That's challenging. So just to reduce this costing and the time, what you do, we check some few parameters with the different clones, right? And we see that which clones can give a maximum yield, because if the yield is more, then costing is less. Always will be interested. And along with the science, actually, in the industry, what happened, along with the science, we also focus about the costing actually, because end of the day, we have to finalize the costing of the molecules, and that we have to buy. So it's all focused from the day 1 to day 0 to day 100, if I consider the 100 days total life cycle. So we consider about the costing. I'm sure actually, that the research lab, personally, I did not focus too much about how much costing of all the reagent. But here, we always choose the quality as well as the costing. So every day we evaluate these two parameters. That's extra pressure, I know actually. Some of the cases, some of the kit, some of the molecules are expensive, but you use for to get a better quality. In that scenario, that's why the biology drug is so expensive. So physiochemical properties, we check. The biophysical characterization, we do. We do some risk assessment, some immunogenicity assay, and some of the formulation screening also. Formulation means after the protein purification, we need some buffer to stabilize the protein. So it's a very small scale study where we understand the protein is stable, and that protein can be stable, scaled up, and that protein can be purified. So that kind of the effort, we generally, the academic lab, we don't give, but industry, we have to, we spend almost minimum 6 months to 1 year to do the durability assessment and the manufacturability assessment. Because generally, the lab, once we start actually the 1 KL to 1 liter, 2 liter, but once it goes to the 1 KL level, the manufacturing, if we don't get scale up in that level, but we just evaluate every stage actually. So then it will be not profitable actually. So then we have to go for the different clones and different molecules. Sometimes we just postpone the enter the R&D of this molecule just because we are not able to scale up. And this is majorly problem actually fusion protein and the MAPS, we did not see too much challenge. But bispecific and as well as the multispecific protein was in a lot of cases that during this study actually you understood it cannot be scaled up. So we postponed the molecule. The molecule can be the good indication, but just because of costing, because we have to reach to the patient. That's a target actually, not the generating the data. Okay, so optimized candidate with the acceptable drug-like properties. So initially we understand at the very bench level, so how it shows the drug-like properties. So this is very critical, the parameter in the regulatory already defined, and the industry also most of the people engage in the CMC section and the CMC division. I'm going to come one by one. So what I'm just trying to say here, this is actually what I can say is the practice. It's a CMC practice, a CMC strategy. So what we do actually, we just take every batch, whatever we produce, all the batch should be identical. It's not like today one protein we purify and tomorrow something we purify, It's not, so for day 1 is showing active, day 2 is not active, is inactive. Then we are in trouble. So this is actually this parameter and this practice and this strategy, this CMC strategy helps us to make the, every day, whatever we start from the, day 1 or after 10 years or 20 years, still the molecule in the market, it shows the same, effective efficacy as well as the potency actually. So that's a CMC. So what it has actually, the different, it ensures the product quality, the manufacturing reproducibility, the regulatory compliance, and the patient safety. So and then next, if I comes to the major component of the CMC, so drug substance is kind of how we reach to the to drug substance. So drug substance, once you purify, whatever you purify the protein in the lab, so that's a drug substance. That's not a drug product. Drug product comes actually after the formulation. So what do you do? So cell line. I'm just coming. I'll just have a one-on slide actually to one by one to give a to reach to the actually how to go to drug substance and then to drug product. So from the drug substance, we add a lot of buffers, a lot of excipients. I mean, you guys also add actually a lot of cases the polysorbate, right? I'm sure during protein purification, we use the polysorbate. We use a different surfactant, different buffer to stabilize the protein. Because proteins, whatever I purify, we have to ensure that this protein is stable. Otherwise, what happens if it's not stable, it is pretty precipitating, then it cannot be injection. Actually, it cannot reach to the patient, right? And next is the biomanufacturing process. The biomanufacturing process is just a manufacturing process. What we do actually here, the drug substance and drug product, we do some R&D actually here to make the DS and DP, and then it's sent to the manufacturing. So manufacturing, there is no R&D. That's only the manufacturing. And that there is also big role play actually the quality control and the analytical characterizations. Every stages, every time we understand actually how the quality of the proteins, and based on the proteins, if the quality is good, then it can go for a DS or DP. If it's not as good actually, then what do we do actually? Then we have to go for it. So that's actually the R&D process, development process. So we take almost couple of years to optimize the process. So that process can go to the manufacturing. So now I'm just coming to the cell line development. I'm not going to very details here actually, but what we do actually, I say the target some of the cells, maybe 3 or 4, the lead candidate we take. So all the 3 lead candidate, what we do, we just make a stable pool generation. First we make a stable pool generation, then we go for a single cell generation, and then we just check whatever the cell line we make, the cell line is stable or not. Because actually we have to reach to the maximum passaging. If you go for a more passaging, it will be cost effective. If the cell is, after 30 cycles, 30 passaging, actually if the cell is not surviving, that again is a kind of challenge. So we always ensure that as much as possible to go for passaging the cell, and the cell line is stable, so then it can help actually, so it can survive longer time. So it's today whatever we make a medicine, it's not going to be kind of only for the 1 or 2 years. It's maybe kind of longer run, maybe the 10 years or 20 years. So we make a cell line, we just store the cell line, and that cell line we always use for the manufacturing. So then we go for an upstream development. So what we do upstream development? Upstream development is just whatever the antibody, we get the best lead, maybe 1, 2, or 3. Generally start the R&D as a 3 candidate. Then we do the different medium optimization to get the maximum yield. So if you get a more yield, that clone can be helpful. If the yield is less, because some of the cases we see actually the yield is more, but the cell is not viable. So in that case, actually, there's a lot of scenario happen. So that's why we always take a 3 to 5 maximum lead candidate to evaluate the end of the process. So after the medium optimization in the shake flux, we go for a bioreactor, and then we just do some process optimization in the bioreactor, and this for the manufacturing. So I'm sure this is kind of quite a familiar in the academic lab. We also use the same petri dish for the cell line development. And then from petri dish to the 2 liter or 5 liter scale up. And from this 5 liter, we go for a almost 50 liter to 2000 liter bioreactor. So, but this is the catch actually. So we generally don't directly go to the manufacturing. We check the scale up actually, right? For initially, we just grow the cells very small scale. Then we go for the 5 liter bioreactor. We see the how is the scale up efficiency and the developability assessment also we do. Every stage, every scale up actually we do the developability assessment to check that how much you are scaling up. As scaling up, after the scale up, the product is stable, product is potent, and it can kind of stable also. So next is the downstream development. So what downstream development? What we do? We just take the cell harvest, and from the cell harvest, we do the purification. So use the different resin for the purification. Mostly for MAB actually we use the protein A column, but MAB is not the only one molecule what we use in industry. There are a lot of therapeutic proteins as well as the bispecific protein. Actually we use the FC domain actually. Sorry, Fab domain. So that Fab domain can be utilized for the first purification stages. There are few chromatography also we do to ensure that the protein is highly purified. So that's the acta purified. I'm sure maybe the lab you have actually, right? That we start from there, and then after we go for a scale up, and then for the manufacturing. So this is the manufacturing, how it looks like the chromatography, but we don't start the manufacturing directly. We just start from the very slow milligram or gram level. Then we go for a 100 gram level, and then we go for a kilogram level. So directly we don't. If you have to go for a milligram, so most of the R&D activity we do the milligram level. Suddenly from the milligram level we don't go for a kg level. So intermediate we take another stages to ensure the product is behaving the similar whatever we have done in the small scale. And that's the formulation development, whatever I told. After purification, what we do, we just add a lot of stabilizer agent like sugar, amino acid, buffer, different buffer scouting we do, the pH and the different preservative also use, and the excipient also, and then filter, and then fill finish, and then go for stability. I'm just going to say something about the stability study and the final formulation. So this is actually the after formulation, we make the drug product. So that drug product directly go to the patient. So here the container closure, why I'm saying actually, I mean, you guys may know, maybe saw actually the molecule, whatever we see the injection, all the injection is not same format, right? Sometimes we give a syringe, sometimes we use vials, sometimes the lyophilized product with a placebo. So there are the different format. It's not the same format every time we use, right? There is some reason. Anybody add actually why is the reason actually here? Right. So that's actually this is a very critical in the stages of how the administration. So after making the DS and DP, we also ensure what is the container we are going to use. If the molecule we are going to lyophilize, we are using this kind of the container. But for insulin, we never use actually this kind of container. We always use syringe. Most of the small molecule, in fact the biology is also used by the syringe. So syringe also we evaluate like whatever the component, what are the molecule we are going to use. So there's a lot of detailed study. There's a lot of data. I'm sorry, I'm not able to show all the data because it's the industry. There's a lot of the limitation to show the data. But this is the activity, the study we do. The container closure system. We have a huge. The department actually they do understanding about how the container closure is impacting. Is it impacting in the stability or not? Anything leaching also coming from there or not? Because this kind of different container, it can leach actually some of the molecules, right? So to ensure that they are leached maybe under the permissible limit. So there are a lot of analytical assay also plays a role in this domain actually. And the fill finish, fill finish is just nothing but the drug product molecule whenever going for in a vial actually. So it's just a sterile filtration, and we do it's a manufacturing practice how the fill finish, and then administer to the patient. So here I'm saying the stability study. I'm sure you guys, whenever you go for a the pharmacy, everybody will check the expiry date. Everybody check the expiry date, right? And we ensure that the molecule should be the expiry date. In fact, for some of the cases, I would suggest actually always you don't buy actually anything very closer to the expiry date. So anybody know actually how we establish the expiry date or the shelf life? So this is actually the study, the stability study. What you do after the drug product, we incubate the molecule in a different temperature, like the 4 degree, like room temperature, -20, or minus 80, whatever the molecule storage temperature, we keep for a 6 months to 24 months, and sometimes the 5 years also, 3 years, 4 years, 5 years, to ensure the molecule is stable. If the molecule is stable actually of certain time, then we establish this molecule can, end of shelf life, may expiry date can be 5 years or 2 years. Most of the cases we for the biologics drugs, we go for a 2 years. But some of the cases we also we see that after 3 or 4 years, it can be used. But that kind of the information maybe some of the industry have actually. But you may not have. So industry sometimes what happens to reduce the costing. If the end of shelf life is more, then costing is less. Right? Then molecule can be, stay for the counter for a longer time. But if the end of shelf life is 6 months, then the costing is high. So this is a very critical study in the forced degradation. So what we do, the molecule we do for the different stress actually we give it to the molecule to ensure the molecule is stable and what kind of the impurity they generate. And based on that we calculate. So in end of shelf life is the mathematics. It's not a simple whatever I showed in one slide actually. It's a lot of the information, lot of statistical model, not modeling actually. It's just a numerical calculation I do. Because of we understand the trending actually all the impurities and the product also, the main product. So yeah, so I think all I covered actually the clone development, stable cell line, and this stable cell line we make only one time for one molecule. So this is keep actually almost maybe 20 years, 30 years actually. Because it's a master cell bank, whatever the MCV and WCV. So master cell bank is a very highly controlled area. Generally, everyone don't have any access. So that's from the one vial we make a working cell bank. That we keep it in the lab. That is also under control. You have to use the inventory. And industry, everything you have to write actually inventory. So everything should be traceable. Then from stable cell line, we go for a cell culture, upstream, downstream, drug substance, formulation, whatever I discussed, and then drug product. So from clone development to drug product, to ensure everything is all right. So if the stable cell line is okay or not, how do you check actually? There will be a lot of analytical testing is required. So to ensure that this stable cell line is generated, like the plasmid, whatever is in there, the plasmid is the sequencing. We ensure that after 30 or 40 passaging, the sequencing is same. There is no mutation happen. So this kind of analytical testing we do to ensure that the stable cell line, whatever we formed, whatever we stored actually is nice, then good condition. Then cell culture. After cell culture, every cells we don't go for a downstream. There are some parameters actually, the analytical parameters are there. To evaluate that cell culture, whatever is grown the cells, it will go for the downstream or not. Maybe the academic lab we do, always we don't consider actually the upstream and downstream because we just grow the cells and go for the protein purification. Then you check the activity. But industry, we don't have so much luxury to do that because it's kind of huge quantity and there's a lot of cost involved. So every stages, every kind of unit operation, we ensure that this is very all quality parameters are fine. Based on that, we go for the next stages. So yeah, so this is actually the kind of then analytical methods absolutely required. All the proteins are different, so analytical method will be different required. And we finalize some specification. What is specification? This is a number. What I'm saying, the 90%, if the more than 90% protein is pure, then that molecule can be taken. If the less than 90, we'll throw the protein. So we'll ensure the 90% protein purification today or tomorrow or after 1 year or after 5 years. Because industry, there's not luxury, whatever the data we're generating, 5 years say actually, no, it's not able to reproduce. So to make it reproducible, we take all the precautions. So they will keep the specification. This specification initially the very early stages R&D, we don't keep it very tight, but at the time being, once you go for a manufacturing, we make it tight. So that because that specification will release, quality control release the product, and that product goes to the formulation and as well as for the patient. So we have to ensure that it is safe, it is potent, and it is effective actually. So this is all analytical tests actually we do. I mean, it's hard to explain all the data. The majorly the analytical equipment, this is the equipment actually kind of plays a crucial role actually for the product release. I'm sure the kind of HPLC. I'm not sure the capillary electrophoresis. You guys may not be using in the lab actually too much, but HPLC might be having actually, right? And then UV spectrophotometer and particle counter, because particle counter is also very important because that is giving the understanding that the protein is not aggregating or aggregating actually, because protein have a very general tendency for aggregation. These are all the tested analytical methods we use to, this is critical quality attributes. Why you saying actually? For the product release. We have some specification, some number, the peptide mapping for the HPLC, if is matching or as per the expectation, then it will be released. Otherwise the product will be not released. It's not like whatever the product we make, we just release for the next stages. And then some physicochemical characterization where the mass spec plays a very crucial role. We have a lot of tests. You can see the lot of tests we use actually by the LCMS. And then SECMOLs, higher order structure, primary structure, secondary structure. So protein, sometimes you can say the primary structure both are similar. One amino acid small actually they change in the so local folding. It can change the global folding and then the overall folding change, activity is gone. It's happened for a very frequent cases. I saw a lot of cases in the academics, absolutely. Industries, multiple times actually we noticed. So that every time we ensure that the protein, the primary structure, secondary structure, tertiary structure, all the quality parameter, impurities, everything well characterized, then it goes for a next stages. So it's not a one or two tests we do actually and say no, it's fine actually and then go for a next stages. No, we test all the tests. We go develop the methods and everything is comfortable, everything is as per the specification and the good quality. Then we go for DP. If anything challenge, either we go back to general what is the problem, we sorted out the problem and then we go for the next stages. It's not like we go for the next stages. And then bioanalytical lab also, the bioanalytical, lot of cell-based assays are there. I'm not explaining everything here, but this is a majorly, we use actually ADCC, CDC assay and the reporter gene expression assay. Host cell protein also, so we also check actually how much host cell protein contamination is there in the protein. In fact, the host cell DNA also. So this is the equipment and the tissue culture lab is required. I'm sure this equipment maybe you guys all read studying in the textbook actually, and the same equipment we are using, like it's nothing the different from the whatever the academic lab, and the data is also similar, but there are some limitations, some control, more under control actually we do all the exercise to go for the next operation. So another is the storage condition. I think the storage condition for the protein drugs as well as the cell gene therapy, whatever the molecules, whatever you use is very important. We always ensure this molecule stable in Y. And any kit actually Y is stable, is a 40T or minus 80. We ensure always, right? This is there or not. So that's the part actually always we very careful actually. And some of the cases we see that 2 to 8 degrees is stable. Some of the cases minus 20 to 80 degrees. And also the physical stress. I'm sure a lot of cases you guys saw actually the protein, if a little bit of frothing you saw actually, right? A lot of foaming. If you do the regular research on the proteins, there's a lot of frothing happen because actually if you little bit of stress, if you protein is getting forming foam, so what happens? The protein starts unfolding, and then its activity is going down. So ensure actually these two factors always for the storage condition. It shouldn't be any kind of stress actually. This is now the second section. Anybody quickly? So which stage ensure the biologic drug is safe and effective in humans? Preclinical testing, clinical trials, process development and manufacturing.
Option D, the manufacturing.
Process development or manufacturing?
Manufacturing, option D.
Manufacturing is later actually. Initially we check actually the process development on the preclinical stages. So basically the preclinical stages we ensure they're safe. So preclinical stage, what we say the preclinical stages is before the clinical. Whatever I just told actually the developability assessment. During that stages we understand the protein protein is safe. So we do all the evaluation study. We don't wait till the manufacturing stages. Purification of the biologics protein, chromatography, PCR, gel and spectroscopy. Chromatography, right. Analytical techniques is commonly used for the protein characterization, mass spec, X-ray, MRI and CT scan. Mass spec, right. So which parameter is critical for maintaining biologic drug stability? Think about the protein stability. Temperature, high pH, sunlight and high pressure. Yeah, somebody is saying no actually. Then what is the option? I heard actually A. Then what is the next? Then A. Yeah, it's a temperature. So any protein actually, kind of very sensitive to temperature. So first you have to ensure that whenever you do the protein purification, everything, you have to ensure the protein, either you have to work in the cold chamber or the protein should be in the fridge actually, I mean in the ice bucket actually, right? So he said, or some of the protein is stable, that can be purified in the room temperature. So we have to ensure the protein, how is the stability. If the protein is stable in the room temperature, you can do all the activity room temperature. If the protein is very high sensitive temperature, you have to, do in the cold room. I'm sure a lot of people use actually, you must be seeing actually that a lot of people are purifying the protein in the cold room. I did actually, sometimes it takes more than one day actually to purification in the cold room. It's painful, but you have another option, right? That's a demand for the protein actually. And which component CMC ensure the manufacturing process consistently produce the product with the acceptable quality? Clinical trial, process validation, drug marketing, and pharmacovigilance. I think this question is a little bit out of drug marketing actually stages. That they are actually, drug marketing actually after the clinical trial, we go for a drug marketing, and this stage is actually the, all the acceptable limit we ensure actually. So regulatory guidelines, I think how much time, Dr. Amarnath, I can get more actually? Half an hour? Okay. Now, I think I'll just, focus the next section wherever I think they might have more questions. But regulatory guidelines, I can be going quickly. So regulatory agency, whatever I told, there are a lot of agencies in the different country, is a different name. They approve the molecule actually. So whatever the molecule we made, now this molecule we have to get approval to go to launch in the market, right? It's not the molecule is not going to sit in the industry. We have to go for a market. Either you have to go for India or Europe or US. So that's a target for the most of the Indian organization. So other is rest of the rest of the world. The rest of the world majorly it comes to all the African countries actually. So that's a target for ours. So first we target the US FDA and EMA. And if you US and FDA, if you make your molecule that specification, it's a very tight specification, then automatically flies to the CDSCO as well as the ROW market actually. You are getting my point, right? The specification I told, right? There's a few parameters of the protein, so that is a very tight actually. We have to maintain that parameters to ensure the molecule is safe and potent. So that's the name actually. I think the US FDA is the United States. The U.S. Food and Drug Administration, so that's the agency for United States actually, and then EMA is for the European, Japan is for PMDA, the Pharmaceutical and Medical Device Agency, and the CDSCO for the India. So any drug approval required from the India, you have to apply for the CDSCO actually. So there are different stages. I'm going to that domain actually quickly, and this agency what they do, they just check the CMC, whatever I just mentioned, the CMC part, enter the upstream cell line development. Analytical methods, characterization, all the data actually they check. The CMC is a very critical, for the industry. It takes a longer time actually to establish all the documentation. Then clinical data, absolutely after the CMC, you have to show that the molecule is clinically active, and the safety also. So there is one organization called the International Harmonization Guidelines. So what they do, this organization, so different country, they have a different kind of guidelines, right? So what these guys actually they are doing, the different enter the world actually, they are harmonizing these guidelines, and they are just making some kind of recommendation. So that recommendation, if you follow, it can fly to the different country. So that's a 5 modules actually we go for the what I can say for the regulatory submission, the module 1, 2, 3. And this slide may be kind of a little bit of interesting. So what we start from the basic research here, and then drug discovery, preclinical, then clinical, phase 1, phase 2, phase 3. Then we go for ND application, license application, and then FDA review, and then ongoing safety surveillance. That's a market. So this is the enter the stage. So one biologics drug development, it takes 10 to 15 years, and for the biosimilar, it takes a little bit of less, but still is a 6 to 9 years. I mean, less than 5 years is very unlikely. It happens for the USFDA market actually. I'm a little bit of skipping some of the slide actually here, because it's all kind of the whatever the year I told, 10, 15 years, how it takes a drug discovery, 2 to 4 years, preclinical actually 1 to 2 years, CMC 2 to 3 years. So CMC part actually 2 to 3 years, and this part actually, if you're ensuring everything is okay, then the other part, then IND preparation, this other rest of the part will be very much clear, because CMC will also ensure the how it could be manufactured. And biosimilar regulatory pathway. So there, whatever I mentioned actually, their analytical similarity is very important. If you show this molecule and these two molecules are very highly similar, so their molecule can fly. And if it's not similar, some of the cases you have to show why it's not similar. If you're able to show, then it's fine. So it's a regulatory review, what they do actually. And then this quiz session, would you like to participate, or I can skip quickly? Okay, I think let's just go. Which regulatory authority is responsible for the approving biologics and biosimilar in India? Option A. Good. Then, which regulatory document format is commonly used for the drug product submission? CTD, patent document, research article, and clinical report. Option? Just loudly. Option A. Yeah. This is good actually. Yeah. Then biosimilars in US are approved under the which section of the public health? I think that it was there actually. It's a 351K, because there is a different pathway actually. You can apply for the what I can say for the regulatory for biosimilar is a one pathway 351K. 351A is for the biologics, and EU biosimilars are regulated by it's EMA. Pharmacovigilance is important for the pricing, marketing, post-marketing, safety monitoring, and the manufacturing. I think I did not discuss too much actually. This is actually the one slide, the post-marketing safety actually. So after marketing actually, kind of once the molecules, so lot of scenario actually, you can see that some of the molecules people actually recalling, because after 1 or 2 years, the molecule have some of the impurities. Recently, one of the molecules happened actually, from the FDA, they recall the product because of they identify the new product that they could not characterize. Some of the new organization they characterize. There is some research article, and they recall the entire product. So this is actually the agency, kind of the surveillance, pharmacovigilance is a department actually, not the agency. There is some agency actually over there, but majorly the organization, they have a particular one department. They work on this, after the marketing, how the molecule behaves. Is there any kind of understanding about any kind of complaint or any kind of the product? So they take some of the molecules every time, just end of shelf life. They take the molecule after 6 months or 1 year, check all the quality parameters is okay or not. So that's their job, and they just make all the databases actually. So I think this is actually kind of my the topics. I think more or less everything I discussed in the opportunities, challenge in the future. But still, I thought kind of I can summarize. So the growing demand of the biologics, I'm sure I have discussed. The expansion of the biosimilar market, that is also discussed. Advanced therapeutics, because the small, there are a lot of scenario we see actually the gene therapy is the only option actually for some of the diseases actually. With a very rare disease, no medicine is available, only gene therapy is there. But this is quite expensive. That's why all the efforts people are putting over there. And the mRNA therapeutics also in a new area where the people are focusing. The country, majorly they investing, where the US, India, China, and South Korea, they are investing a lot of money for this, biologics therapeutics. The challenge in the biopharma industry is the high developmental cost. So this cost is too high actually. That's what I'm saying actually from the day one, we think about whatever the reagent, whatever the molecules, whatever we take, everything is accountable for the costing. And this clinical trial. Clinical trial is also very expensive, and the clinical trial, the different country like the US, it depends on different subject, how many patients you are going to consider. So depending on the costing is also high actually. So based on that, we also sometimes consider let's just focus on the Indian market or RO market, or go for EMA, or go for Europe or US actually. So that's a challenge. Still, I mean, we are not so much rich yet to invest the money for the clinical trial actually. And the molecule development cost is almost 2.5 billion. And the manufacturing is very complex. A lot of scenario we see that during the manufacturing stages is failing. It's failing like 2,000 liter liter stages once we purify. Well, because we see the 5 or 10 and then 200 the intermediate, and then for 2 kL we see the manufacturer 2 kL if he's failing, think about the 5 to 7 crores loss. Right. So it's a very complex kind of challenging we see actually, because it's a protein is always complex molecule. So scaling up from lab scale to manufacturing is not an easy job. The way actually I'm presenting here is not so easy actually. It takes almost, I showed 10 to 15 years actually for one molecule. So regulatory complex. So regulatory also they are very highly focused on the CMC part as well as the safety and efficacy, because whatever the molecule you are injecting, we have to ensure that it's a safe, and then shows the efficacy actually. And another thing is supply chain, because still I feel we have a challenge to one country to other country, but one place to other place, the supply chain actually. Like, the protein, if you stable in the 4 degree, I mean, how can we just bring the one, it's not an ice bucket actually, right? We bring from one lab to other lab. It's a kilogram protein, right? So we need a big bucket actually, kind of the minus 80 or 4 degree temp. 4 degree is kind of manageable. Some proteins are minus 80 or minus 20 stability is a real challenge for one place to other places. This is a challenge in the biopharma industry. Future in the biopharma industry, I mean, so far the artificial intelligence, I'm sure everybody is somehow, I also use some of the ChatGPT, I see a lot of students also they are using. So there is there's no other option. We have to use the AI for every sector, and drug discovery. So far the developmental part we are using in the limited way, but there are a lot of the activity. It's a very early stage I can say. But discovery stages, the target identification, the protein structure prediction is helping really a lot. So far, if I say, kind of drug screening in the discovery stages, the AI already made a significant progress. Because a lot of data actually initially, right? You go for 1,000 clones or 1,000 targets, and a lot of data. If you use the AI, actually, it can, kind of, what I can say, ease your life actually. And continuous biomanufacturing. So continuous biomanufacturing, it's a big chapter in the manufacturing process, because what we do, which we actually one molecule we do in the one facility, one for one manufacturing. But continuous manufacturing, what helps actually, it can one places we can go for a few molecules for manufacturing. So that's people are some of the industry, they are investing money to do that, because if you go for 4 or 5 molecules manufacturing simultaneously, or automatically reduce the costing. So future of the biopharma industry is personalized medicine, as well as the growth of biosimilar. I mean, if you do biosimilar, we can reduce the costing. It can always help actually for the because whatever I see, most of the Indian companies, they focus on the biosimilar, because it can be a little bit of can be a little bit easier to to go for a market and to help for the people actually, because if you could reduce the costing of the price, it can be achieved, accessible to the common people actually. So that's the main target, and that's the main the area for our business, main area for the people opportunity also. So now actually kind of I categorize. I think it can be a little bit of helpful for the students here who are really interested to explore in the industry. So this is all the area whatever I discussed. This is a division or department, you can say. You can always kind of explore your expertise. I mean, initially you may not have expertise, but you can go. This is a domain wherever you can go for an internship, as well as go for a next level. Once you go for a little bit of understanding, then you can grow that domain. But that's a total domain. Actually, so many domains are open, and the next 5 and 10 years, I can say, is a huge demand in biopharma industry because of the kind of the market is so far expanding. Number one. Number two is actually the lot of rare disease, as well as that are very very less side effects. So a lot of things I actually already explained. So here I think you guys can think about in future, to explore. I mean, I'm sure it's all of the words. It did not 100% reach to everyone, but try to say. But I would kind of happy to get the questions. But this is actually the new whatever Dr. Amarnath was mentioning here. So Biovignana Biopharma is my new initiative actually, wherever the last almost 6-7 months, I am working actually as a consultant, as an individual consultant, where I'm working actually different company to majorly the CMC part actually, the cell development to regulatory part, different part actually, and also I recently this is all the based on this whatever today's discussion and some of the all additional some of the few things actually I thought it can be kind of make as the courses or kind of the and also working with the academics and industry also. So it's right now is a very early stage, but then after what I'm just trying to talk with the other industry partners actually. So what they are maybe somebody is I'm actually basically analytical and CMC expert. Somebody is good in manufacturing. They can give a more understanding manufacturing. I never worked in manufacturing. I mostly worked in the different stage of R&D actually. R&D level. So R&D part I know. So that's how actually the idea to the consulting and training the organization. And this is the 6th module actually so far we have designed. There are some few industry experts also they help later where we can cover this all the 6 module. It can be it's not decided actually, but some of the cases on-site classroom also actually training or workshop can be arranged if some of the university or some of the but this is typically actually this slide for the academic organization, not for the industry. I'm also kind of conducting training for the biopharma early stage industrial professional. For them, it's a little bit of advanced stages. But for this is all the basic things. So there we will discuss not in the one day. It can be a little bit of 10 to 12 weeks minimum, or not weeks, maybe session actually, maybe the 6 to minimum 6 weeks actually. There we will be discussing actually all the biosimilar biologics and how and all the important. I think this is a very important part, the career guidance. And whatever is the expectation from the industry, so what questions, whatever the understanding, then at least they expect from the textbook knowledge. They don't expect actually you should have an understanding of the biosimilar biologics, but at least you come with some understanding, some of the things actually. You know the proteins, but how the proteins can be utilized as a drug. Nothing wrong to learn actually, right? So that's a Biovigyan Biopharma initiation. It's very early stage right now. I'm saying, and maybe it can take a few months also from today. And next is a question answer. I'm sure the people are tired here, but I'm happy to take the questions from the students, faculty members. No questions? But if you have any questions, like you can always reach to it's my contact information. It's not possible to give it to all the everyone to my information. So you can take a picture or just write down my email ID or phone number to write down the what is exactly what can be done actually if you're really interested about for the biopharma industry. It's not.
Hello. So like you just said that we can go in the biopharma industry after our degree. So like after BSc or masters, like how can we join the biopharma industry?
Right. Now see, right now the BSc, so we don't differentiate the BSc, MSc, in fact PhD. So we all consider as a freshers, right? But expectation is a little bit of always higher actually. If you're a master student, we expect actually you are mastered in some of the domains. So once you go for a this kind of first I think you should try actually for the internship program. The way actually the internship program the students attended for the different country for the academic program. So you should attend actually the industry program. So once you go for 2-3 weeks the industry, or maybe sometime we take actually 3 months or 6 months also internship as a BTech and BSc masters as a project actually. We always not always you have access to the everything, all the data, because a lot of data are confidential there. But you will get to know actually what's going on there. I mean, it's a small part. Generally, we assign to the junior level. The students, they come actually. And a lot of scenario we see actually, students are adding a lot of values that I'm not able to think about. Or maybe the experienced students actually, the industry professional, they are not able to think about. So students always give, add a lot of values, whatever I see in the industry. So your target should be, kind of whenever if you company cell and gene therapy, at least you go for some of the understanding of what is cell and gene therapy. If the biosimilar biologics industry, maybe most of the cases the biosimilar industry you can see. At least what is the biosimilar? So that the basic things at least we can expect from the master level student or bachelors. If the PhD somebody can understanding about the if you see actually, everywhere, the different department, if you have a, cell line experience, then you can go to the upstream and cell line development. If you have protein expression, protein purification, you can go for protein purification. If you go for a mass spec, characterization experience, you can go for characterization. There's a different department. In fact, additional some of the regulatory departments is a very crucial role they play actually. All the data, SMEs, they give it to a regulatory team. They combine, and then it comes for all the kind of senior level for the revision. I was a part of some of the regulatory submission actually as a review. So that's another domain actually. You have to know the science actually. So it's not mandatory the BSc and MSc and PhD, but at least you have to show your interest that you are that you want to. I mean, if somebody is calling for an interview or internship, you just try actually over there. There is a lot of position. I don't know about the Kolkata may not be having too much organization, but there are huge organization in Hyderabad, Bangalore, as well as Pune, Mumbai, and they take a lot of students. In fact, the Biocon, Dr. Reddy, Enthas, Dr. Reddy and Biocon, Syngene, Origin. In fact, personally, I had a bunch of the students over there as an internship, as well as freshers. But freshers, directly you cannot get an employee. You have to go for some training model over there, either internship or some of the cases, technical training. So 1 year or 6 months, like probationary. If you're really doing good job there, we hire actually. Right. Most of the cases we hire actually, because the students are always, adding a lot of values. Right. It's just a mindset actually, right? I know that after BSc and MSc, I also had the same mindset actually. We should try, start the PhD actually. But I nowadays I see in the last 4 or 5 years, I think, there's a lot of students coming in the industry. Oh, is it? Right. Now, Dr. Reddy and Biocon, they are focusing. Biocon is, if I consider the 10 years before, it's typical academic lab. They, always encourage to that's why Pfizer, Biocon and the Reddy. In fact, I also started from the Pfizer actually, after postdoc actually. They entertain, the postdoc level, but absolutely, early stage career is always easy to get a chance actually. Any department, if you go the junior level, is a lot of students, they are there actually. But once you PhD, little bit of limitation, postdoc, more limitation, right? After postdoc, I think, there's no other scope to go to the industry. But a lot of cases, actually, I think US, I saw actually a lot of cases, they switch academics as well as the industry. That I see actually. Here, is the possibilities there. It's not like the, the academics to industry and industry to academics. A lot of cases happen. And every initiation, every talk, wherever I go to the university, I always say actually, and I try to link. And this organization, whatever I'm making actually, that, idea to work with the industry and academics both. The partnership and the collaboration. So we'll see, how much, I can achieve over there. But this is actually, the things at least, if you at least, go for a Google, ChatGPT, you write actually what is the biology, then you get a lot of information. Learn actually and then go for interview, and then I'm sure it will be opportunities there. And this is a good progress actually, It's a lot of opportunity, lot of progress. You can, grow actually in industry also. No, see, I think that's a early stage whenever we take as an internship the students. So sometimes we induce the student in a different department. It's like in the PhD, whatever the people go for the different lab to understand. Here also we go for a different, we assign for the maybe 2 or 3 weeks for the analytical, 2-3 weeks for the upstream and downstream. So based on their interest, we try to accommodate there. As an individual, I tried a lot of effort actually in that way. Actually, what happened? The industry and the students both can be benefited actually. But sometimes you don't have any option. You just go there and you go with the flow. That is some of the cases there actually. But some of the cases actually, like the Dr. Reddy, Biocon, if you go for the big organization, and particularly if you go to the startup, there are a lot of startups actually there, the Bangalore and Hyderabad. There is a lot of opportunity to learning. Yeah. Startup actually, you have a lot of opportunity to learn actually. You have to do, a startup is like a academic lab. So there is a lot of opportunity to explore a lot of techniques. Because if we go to Biocon, there's a one department, 100 people are there. But if you go to the startups, if you say that 10 people are or 20 people are running the entire function, so you'll have a lot of opportunity to learn actually. So it's a startup and Biocon, always both have pros and cons are there actually. Right? So that's how the decision actually. I mean, people after masters it's really hard to understand which domain. But after that's why like if you know some of the understanding about that the department, then you have to take a decision actually. Yeah. And there is a chance to change actually. So a lot of cases the quality assurance, like the business development and the program management, this kind of the team. Initially 3 to 5 years people incubate in the technical domain and then they go. It helps actually. Otherwise if you are selling something, if you don't know anything, is a problem. So the business development, when somebody comes actually, they have to understand actually kind of some of the molecules. Other is how can they sell. In the project also the management as well as the quality assurance and the regulatory. So this is all the domain we are expecting at least somebody incubate minimum for 3 to 5 years. Then they come to this domain. They can add actually a lot of values there. Because it's majorly the supporting department actually. Yes.
Good afternoon, sir. My question is that as you mentioned earlier about the internship program and all, and also you mentioned that about the training program. Right. So how can we enroll into that, and is there any exams that we need to
No, there is whatever I mentioned, this is very early stage, under registration. But I think this is the number actually. If you the email actually, if you and just follow the LinkedIn. If you follow the LinkedIn, just keep in touch with me, and it will come very soon actually. Within a month it will come actually. Okay.
Right. Thank you, sir.
I'm just waiting for some some administrative work. Once it's done, then it officially will be launched, and then it will start. So more or less the modules. Things are almost ready, but if the university, if you guys interested, so it's along with me actually another a couple of more people can come actually, with me actually. They are all, different domain expert. They can come actually, if you have some infrastructure, so they can, conduct 7 days or 2 weeks, workshop program actually. Right? So that's a customized. If you, if you say that this is the infrastructure you have, we can come for 7 days and do for a workshop, morning session theory. So theory, then theory will be a little bit more. So and that kind of workshop will be a little bit very topic specifics, not the very wide. This is actually the overall understanding actually overall, right? So enrollment part, I would say right now, just wait and see the look for the LinkedIn. If you write my name, actually you'll get it. Just and then also you can write to email to my personal email ID, or just WhatsApp. Both numbers are in a WhatsApp. So it will live actually soon within a May.
Okay, so good afternoon, sir. Sir, my question is actually, you mentioned that there is often a big technological knowledge gap between academics and the industry. Like the industry is like applying the knowledge of biosimilar, which you talked today, right. But we may not so be very much familiar with that concept in our academics. So I want to ask you, like as a fresher, how should I pursue these technological knowledge gaps so that I can close these things and apply with a good prior knowledge which you talked about?
I think it's a good question actually. So kind of this initiation actually overall, the idea to reduce the gap actually, how you can know actually, like if that's what I was trying to understand from the your faculty members, that how much you covered actually. I mean, it's not possible to cover everything because whatever the information, that information is all our, my personal experience. Absolutely, some of the things are available in the different reference paper here and there. I, personally feel actually this topic is the biotherapeutics part, and all the university should, a little bit of more, add actually this topic. I think Dr. Amit is also here actually. Maybe you can think about actually in this, because what happened, like, if the course is more enabling for the students, for the academic and the biopharma. I'm not saying that, as a biopharmaceutical person, I am not, kind of saying that biopharma is only the options. It's not like that. But people have we should have both option. So if you induce in the student like the MSc and BSc, it can help actually. It can help. Because whenever I see now, some of the university, they have actually the biotherapy. I think the Manipal University already started. I was supposed to go actually one time there. I could not, just because of other reason I could not go. I did not get approval actually to go there. Because in industry, there is a lot of challenges sometimes to attend actually this foreign program. So they have a biotherapeutics department, and there they are, they have an internal, faculty member, as well as they also, inviting actually from the industry people actually. Industry, there is a scope actually. If particularly somebody from the manufacturing, you can always ask them to come here and, discuss something on 7 days. I'm sure the 7 days, if I discuss this thing on 7 days or 15 days, it will be all helpful. And a lot of information I get to do in a 1.5 an hour. Most of the students lost. I see the 5 question out of everybody's answering is very good actually, right here. So that's how actually we can, that's an initiation. I think that's the government also initiating for the NIPER. But this position, whenever we, hire, is not limiting only for the pharmacy. So we consider the pharmacy as well as the bioscience actually. It's not biotech only, the bioscience. Because a lot of microbiology assay actually I did not discuss today that we do as a safety. I talked several times the safety, right? It's very important. So microbiology is the one department dedicated where the microbiological assay to ensure there's a safe. So I think that's how actually if connecting the syllabus or some of the cases like this kind of the academic industry, some of the discussion if you organize, it can helpful. It can helpful to the students. I mean, for you to take a decision, it's not to encouraging to go to biopharma industry as well as, you always have an understanding because academic lab, actually, whenever you go for the MSc practical lab and the PhD, you see the academic lab, how it works. Technically, there is no difference. As I say, scientifically, there is no difference. It's just the way we are managing the science. Because that science, the academic team, in fact, I also did the same thing, they published the paper. But here, the publication is not so important here. I also tried sometime. I have some couple of publications with the collaboration with the academic team actually, because the industry generally don't allow to, to publish directly. But along with the publication, here the target actually, the molecule, whatever you're making, we have to go to the patient. So that's the strategy and the pathway you have to decide, right? And the different molecule protein, every protein is different molecule is different. So there is a lot of challenges, a lot of complexity. Is there actually. There's a lot of learning scope is there actually. I can say there's a lot of learning scope. But if you say the protein science, absolutely it's the same science actually. There's no difference in science. There is the once you academic lab, all the primary structure, secondary structure, quality parameter, HPLC analysis, all the analysis we do in the academic lab and the industry. But some of the practical assay, I don't know how much it's covered. Some of the assays, if you include in your syllabus, that can be helpful. In fact, spectrophotometer, I'm sure you have actually, right? But the spectrophotometer, you use some of the protein molecule. At least if you use some biomolecule, like the insulin is available in the 300 rupees in a counter. So if we consider, that molecule for at least it will be interesting how it, behaves as an insulin. Like circular dichromosome, how it looks like, how is the mass spec. So then it will be, a little bit more connecting. It's not possible to, induce everything to whatever is going on in industry. But at least some of the understanding you can get it from here. The small program also, it can happen in a academic lab.
Thank you.
Same lab in the industry actually. We also stay in the lab actually. Nowadays not, but before 6 months I was there also. Any further question? Thanks a lot, yeah. It's a big opportunity for me actually whenever I interact for the students here. If at least there are a few students come to the biopharm industry, I'm sure the molecule quality will be better and it will be shorten the times.
Thank you so much, sir. It was a wonderful interaction. Now I would request Dean, sir, to come upon stage and felicitate our guest speaker of the day. Thank you so much. Now I would request Dr. Nivedita Banerjee to come and conclude the sessions.
So now I would like to take this opportunity to express my sincere gratitude to all those who participated and contributed to successfully organize this event. I extend my heartfelt thanks to our Honorable Pro-Chancellor, Sir, Professor Dhrubajyoti Chattopadhyay, Honorable Vice-Chancellor, Professor Anupam Basu, our respected Dean, Professor Amit Sarkar, Assistant Dean, Professor Amarnath Mukherjee, Heads of the Department, Dr. Sudipto Saha and Dr. Fatima Kalkatawala, Biotechnology and Microbiology. A special note of appreciation, of course, goes to our distinguished speaker, Dr. Shobhoshachi Haldar, for sharing his expertise and enriching the session with his insightful considerations. Heartfelt thanks goes to Dr. Surjit Singh, Assistant Professor, Department of Biotechnology, for meticulously organizing this event. Also, I would like to thank all the faculty members, the staff, especially Dr. Shauri Roy, Smita Shaw, for their immense contribution to the successful execution of this event. And I definitely congratulate all the students, presenters of their excellent work at Japan and their enthusiastic participation. So also thanks to Dr. Shayani for emceeing this event throughout. And so with this, we come to the end of this session today. And thank you all for being here. Thank you so much.
Thank you, Abby. Now we will show you a QR code on the screen. So you have to scan it and give the feedback. Then only you will be getting your certificates.